that you can share. I
Fine.
will start the introduction.
It is visible.
Yes, sir, it is visible. So, good morning, one and all. Those who have gathered in this virtual platform for this refresher course. First of all, I extend my warm and esteemed welcome to our esteemed expert, scholarly expert, Professor Nandakishore Karade, sir, our course coordinator, Professor Manisha Kodape, madam, and all the learned participants, those who have gathered from the across country to this refresher course. First, we will take the brief introduction of our today's expert, and then we will start with this session. So now may I request Dr. Bhaskar Nikam sir to first introduce our guest, and then we will start with this session. Yes, Dr. Nikam sir, please.
Yes, sir. I'm audible?
Very much clear, sir.
Yes, okay. Thank you, sir, for giving me such a wonderful opportunity. Respected course coordinator, all the members of this organizing committee, and very much dedicated and cooperative technical team, distinguished participants, and our honored resource person. Good morning and very warm welcome to everyone. It's my immense pleasure and privilege to welcome you all for the today's session of this refresher course under the Mission Malviya Teacher Training Program in Chemistry organized at Sant Gadge Baba Amravati University. Today, we are truly honored to welcome our distinguished resource person, Dr. Nandakishore N. Karade sir. He is a professor of organic chemistry and head of Department of Chemistry at the Rashtrasant Tughluji Maharaj Nagpur University. Dr. Karade sir is also a fellow of Maharashtra Academy of Sciences with over 27 years of postgraduate teaching experience. He has guided 12 PhD research scholars and completed 7 major research projects funded by DST, UGC, and URPS with the cumulative research grant of 2.35 crore rupees. He has contributed significantly to the field of synthetic organic chemistry, particularly in the applications of hypervalent iodine reagent. And sir also published 51 research papers and 1 book chapter and hold 3 patents. His outstanding contribution to education was recognized through the Best Teacher Award of Ramanand Tirth Nagpur University. His Google Scholar profile reflects 1,138 citations and H index of 20 and an I-10 index of 33, highlighting the impact of his academic and research excellence. Dr. Karade sir is also widely appreciated for his exceptional engagement with the NPTEL, having completed 16 online certifications, including 11 topper ranks, 6 gold medals, and 9 silver medals. Sir received NPTEL Superstar recognition twice for securing the highest marks in the more than 3 online certificate courses. In university administration, sir has served as an IQAC coordinator or IQAC director, member of the management council, member of the purchase committee, and chairman of the board of studies of Autonomous Department of Chemistry. Sir has also made noteworthy contribution as a national level subject expert for confidential examination and other competitive bodies. Sir is highly regarded as a researcher, teacher, and academic leader. Dr. Karade sir continues to contribute actively towards advancing chemical sciences and higher education. Sir, your remarkable academic journey, dedication to excellence, and commitment towards the future generation, inspiring all of us. On behalf of the course coordinator, organizing committee, technical team, and all of the participants of the refresher course, I extend our heartfelt welcome and sincere gratitude to you for kindly accepting our invitation and sharing your valuable knowledge and experience with us. So I also extend warm welcome to all the participants. So once again, a very warm welcome, sir. Thank you. Thank you very much.
Good morning to all. At the outset, am I audible to all of you, ma'am?
Yes, sir. Yes, sir. Good morning, sir.
Good morning, sir.
Good morning, sir.
Good morning,
Good morning, sir.
Good morning to all. So at the outset,
Morning,
I'm very
sir.
much, good morning. At the outset, I'm very much thankful to the coordinator, Professor Kodape madam, for giving me this opportunity to deliver a lecture in the refresher course. Every year, this university organize this kind of the activity. So for that, I really congratulate Department of Chemistry for organizing such event.
Thank you.
Today, I'm going to deliver a talk on biosynthesis of terpenes and terpenoids. This is a part of curriculum of many state universities. Perhaps it may be the part of undergraduate program as well as a postgraduate program. And if it is suppose not in the curriculum, I recommend to all the teachers that they should incorporate this into their curriculum. Now, before understanding the biosynthesis of terpenes and terpenoids, we will try to know about the basic terminologies involved in this particular topic. These terpenes and terpenoids are actually natural products. Natural products are the compounds which are synthesized or which are prepared by the living system like plants or the animals. The plants and animals produce these chemicals are mainly contain carbon compounds, and therefore we may call them as a organic chemistry. We may call as a chemistry of organic compounds. Now these compounds prepared by the plants is of two types. One is called as a primary metabolite and second one is called as a secondary metabolites. The primary metabolites like the amino acids, then the protein, then nucleic acids, and the carbohydrates. All these are very much important for the growth of an organisms, and they are specifically called as a primary metabolites. The second category is called as a secondary metabolites, and these secondary metabolites are specifically called as a natural products. Now, these secondary metabolites are not directly involved in the growth process, but they are supporting somewhere other way the living systems. So, terpenes and terpenoids are the secondary metabolites. As far as their origin is concerned, mainly they are synthesized from the plants. Nature is very systematic. So more than 90% or 95% compounds in the nature, particularly belonging to the class of terpenes and terpenoids, are synthesized by the plants. Now, the nature is very systematic. It is producing an array of large number of compounds called as terpenes and terpenoids by using a 5 carbon atom building block, and it is called as isoprene. Now, when it contains only carbon and hydrogen, it is called as a terpenes. And when it contains oxygen, it is called as a terpenite. It does not contain nitrogen. Nitrogenous compounds are called as alkalides. So we are having two kinds of the things. One is a terpene and another is a terpenoids, and they are building blocks. They are prepared from isoprene molecule as a repeating unit. Terpenes contains only carbon and hydrogens. Terpenoids contains carbon, hydrogen, and oxygen. Now, more than 23,000 terpenoids are known in the universe, having wide range of structural diversity. We will take some of the examples. Here there are 5 examples. The first example is a myrcene molecule. It contains 10 number of carbon atoms and it is a part of tamalpatra. That is a bay leaf. So bay leaf is used in making a curry, and curry is delicious because of the ingredient called as myrcene. Second one is alpha-phelendrine. Alpha-phelendrine molecule is found in the eucalyptus trees. The pleasant smell and the medicinal properties of eucalyptus oil is due to the presence of alpha-phelendrine. Then in a cosmetic, we always come across the rose oil, and rose oil contains a ingredient called as a geraniol. Then mint, in the mint leaves, a ingredient present is called as a menthol. The pleasant smell of the cardamom is due to the presence of a chemical called as a alpha terpenol. So these chemicals are very much around us. In these structures, what you have to observe that they contains double bond mostly. Of course, alpha menthol is a saturated compound, but many a times it is observed that terpenes, that ENE, this is a generic name. This terpenes name actually it contains mostly the double bonds, but there are some examples where it does not contain a double bond. Let us have again few more examples. The caraway seeds. Caraway seeds contains carbon. It contains a ketone functional group. Then zingiberine. Our morning tea starts with the ginger tea, and ginger contains a ingredient called as a gingiberine. Now it contains 15 number of carbon atoms. These two structures are the bicyclic structure. This is a monocyclic structure, this is a bicyclic structure. In this bicyclic structure, there are two important chemicals. One is alpha pinene and the beta pinene. This alpha pinene and the beta pinene were obtained from resin trees in the African jungle, and they were called as a turpentine. So the generic name of terpene was initiated from turpentine. Another one is a citral molecule. In citral, we are having a aldehyde as a functional group, and this is present in lemon. Then carrot contains a long chain compound called as a beta carotene. Beta carotene contains total 11 conjugated double bond, and it's a very pleasant red color that we observe in the winter session when the carrots are very much around us. Now, from all these examples, what I want to convey you that we should make first of all comfortable ourselves about the structure of these molecules. Once we understand the structural pattern of these terpenes and terpenoids, we can really understand the biosynthesis of all these molecules. What is observed? Observed is that they may be the acyclic structure like myrcene, like geraniol, like squalene. So it can be the acyclic structure. Monocyclic structures are also possible. Bicyclic are also possible. Tricyclic. So there's a wide variety of the structures present in terpenes and terpenoid family. Then if it contains only carbon and hydrogen, we call it as a terpene. For example, myrcene, alpha-phenandrine, limonene, and the beta-carotene, all these are the examples of terpenes. The oxygenated form of terpenes will be called as a terpenoids. Now, the most common functional group in this terpenoid family is alcohol, aldehyde, ketone. These three functional groups are very abundant. Acids and the esters, we will find very less kind of the functionality present in this class of the compounds. So, alcohol, aldehyde, ketones, we are having here a primary alcohol, here is a secondary alcohol, and we are having a tertiary alcohol. So, all these functional groups are present in terpenes and terpenoids. Now, as already declared in the beginning that there is a repeating unit present, that is isoprene. Now, depending upon 1 minute, I had an urgent call. 1 minute, just. Hello. Good morning. सर माझा क्लास चालू आहे. मी 12:30 नंतर फ्री राहील. हो, उघड आहे. प्लीज. Yes. Sorry. So what we have seen so far that the terpenes are made up of isoprene units. It contains 5 number of carbon atom. Depending upon how many isoprene unit contains, these terpenes are classified into monoterpenide, diterpenide, triterpenide, and tetraterpenide. So for mono, we are having 10 number of carbon atoms, that is 2 isoprene unit. Diterpenide 20, triterpenide 30, and the tetraterpenes, that's a 40. 1.5 is called as a cis-Q, so it's a cis-Q terpenide. 2.5 is called as a sister, it is called as a sister terpenoids. So all these molecules are in the multiple of 5. So nature is very systematic, and corresponding to that we are having various examples here, and as we have already seen in the previous slides. There is one important terpene, it's called as a polyterpene, or you may call it as a natural rubber, or you may call it as a resin. This is again nothing but the polyterpene molecule. It's a terpenoid molecules. So from this, what you have to understand is the monoterpene, diterpene, triterpene, and the tetraterpene, and their corresponding number of carbon atoms, and how many isoprene unit contains. This understanding is very much important when we will talk about the biosynthesis of the terpenes and terpenoid molecules. Now, if we look at the structure of isoprene molecule, this is its saturated analog here I have shown you. Is a branched site and another one is unbranched. This branched site can be considered as a tail, head, and unbranched can be considered as a tail. Right. So I have just shown here a one cartoon where you will realize that a branch portion can be called as a head and unbranched portion can be called as a tail. So theoretically, there are three possibilities of joining of one isoprene to another to get a monoterpene, diterpene, triterpene, tetraterpene, and so on. Because these molecules are formed by combinations of these isoprenoids. So theoretically, there are three possibilities. One is a head to head, tail to tail, and head to tail. Now, out of these three possibilities, theoretical possibilities, what is common is a head to tail joining. This is very much abundant in the living system. Tail to tail occasionally we get, but there are hardly any molecule which contains head to head linkages. Now, why we get here head to tail or a tail to tail linkages, that we will see in the biosynthesis of molecules, biosynthesis of terpenes and terpenoids. Now, 100 years back, chemists were not having sophisticated tools, and to understand the structure of these molecules, several scientists have contributed. One of the great scientists, that is a Wallach. Wallach has made a great observation that terpenes are made up of the isoprene units. And for that, there are two evidences we have. One is the empirical formula of terpenoid is C5H8. That is one of the evidence in support of this particular fact. And second one is the thermal decomposition of terpenes gives isoprene as one of the products. So this particular observation was led to a rule called as the isoprene rule. Isoprene rule states that terpenes are made up of isoprene units and they are joined through head to tail linkages. Now let us make a practice, or let us realize ourselves that it indeed contains head to tail linkage. Here example of acyclic monoterpene. It contains two isoprene units. You may ignore the double bond as well as the functional group, only focus on the carbon. We will observe that one isoprene unit is joined to another isoprene unit through head to tail linkage. Similarly, for monocyclic monoterpenes mean 10 number of carbon atoms. So this is menthol and alpha. Again, we find head to tail linkage. In the beginning, one isoprene gets connected to the another isoprene. We get an acyclic structure, and through the enzymatic reactions, it undergoes a cyclizations. That part we'll discuss in the subsequent slides. In a bicyclic terpenes, again the isoprene rule is observed. Here, head is connected to the tail. Then again, we can give this kind of the exercise to the students also. If you are teaching at your workplace, you give them to trace out the isoprene unit so that they can have a better understanding of the topic. A student should at least remember 12 molecules of terpenes, which are used in a everyday life. And if you understand the isoprene rule, then it is very easy to memorize it. Cis-Q-terpene contain 15 number of carbon atom. It means 3 isoprene units are there. And here it is shown. Here it is also shown. Head to tail, head to tail. There are 3 isoprene units and therefore 2 head to tail linkages are observed. In this zingiberine molecule, again the same thing occurs. And in tricyclic terpenoid, cis-Q-terpenoid, that's a santonin molecule, which is very important from photochemistry point of view. We get here 2 head to tail linkages here. Then let us take an example of phytol. Phytol is present in chlorophyll molecule. In chlorophyll molecule, there's a side chain. See the importance. It is not directly a primary metabolite, but its importance is there because chlorophyll is useful in the photosynthesis process. Capture of CO2 getting converted into glucose. So this phytol molecule contains 4 isoprene units, and C is a very systematic way, head to tail, head to tail, head to tail. Vitamin A, which is responsible for vision of an eye, it undergoes cis-trans isomerism. It requires, we eat carrot, and this carrot is enzymatically cleaved into vitamin A. And that's why the doctor recommends you to eat more and more carrot. So this vitamin A contains 4 isoprene units, and again we observe that there is a head to tail linkages. Now we have seen many cases head to tail linkages. Exceptions occurs. Exceptions occurs in case of triterpenoid and the tetraterpenoid. Triterpenoid means 30 number of carbon atom. Tetraterpenoid means 40 number of carbon atom. In a triterpenoid and a tetraterpenoid, It also contains head-to-tail linkage, but in the central portion of the molecule, just highlighted here, in the central portion of this molecule, tail-to-tail linkage is observed. Right. So we are having here head-to-tail as well as tail-to-tail, but we never come across any head-to-head linkages as far as this isoprene rule is concerned. Again, let us make an example here with squalene molecule. Squalene molecule is responsible for biosynthesis of cholesterol molecule, and it is a very interesting molecule. At the central portion, again, there is a tail-to-tail linkage, but in the remaining cases, we are having head-to-tail linkages. So it is my appeal to all the teachers here that if you are teaching this topic, make it more interesting by giving them such exercise to the students for tracing out head-to-tail and tail-to-tail linkages in a wide range of compounds, so that students will understand the structural aspect of this terpenes and terpenoid molecules. So this is beta carotene. The next one is a lycopene. Lycopene is present in a tomatoes. Right. So this way I have explained you now the definitions of terpenes, terpenoids, carbon hydrate functionality, and then we have seen how tracing of this isoprene can be done. Now let us see the importance of these terpenes and terpenoids in the living system. As already I have explained you that vitamin A is responsible for vision of an eye. This is a central double bond which undergoes cis-trans isomerism, and therefore there is a perception of sight. We can have a sense of sight because of this particular phenomenon. Again, another example is of chlorophyll molecule. The side chain present here, this particular side chain, is nothing but the phytol molecule, and that's why it is important. Now, a very famous chemical that is called as a paclitaxel, or it is also called as a taxol, and this taxol molecule is used as an anti-cancer agent. Many syntheses of taxols are available, but its isolation was done at America. And there is one Indian chemist in that discovery, that is Professor Mansukh Chand Wani. Mansukh Chand Wani passed away last, I think, 3 years or back or 4 years back. I personally met Professor Mansukh Chand Wani. Mansukh Chand Wani was responsible for the isolation of this compound from the natural plant. Of course, he worked at USA there at that time. He's from Nandurbar. So this particular molecule is also work as an anti-cancer, and you see here there are 20 number of carbon atoms. So it is also having some medicinal properties. Then if you look at this particular list, nowadays essence means an aroma industry. Aroma industry, main 40% component of the aroma industry is terpenoid chemistry, is a terpene chemistry. And essential means essence. It is having certain flavor. It is having a pleasant odor. And you look at all this: sweet lemon, limonene. Limonene is present in a Mosambik, that perhaps lemongrass, we can make a tea of that. Coriander, our curry is delicious because of the coriander. Pine trees, turpentine contains this. Cardamom, bay leaf, eucalyptus, mint leaves, rose. So they are part of our everyday life. And as an organic chemistry teacher, you should inculcate interest among the students for this particular topic. It is a very fundamental topic. Then, as already mentioned here, this aroma industry is having total share of 36.88%. And this is a very old slide. It's a market of 2016 market share. So terpenes are having a lot of commercial value also. If you go to the hill station, I had been to the Ooty long back, and at that place I have seen the isolations of eucalyptus oil, or some plants were there which are contributing to this, contributing to the terpenes and terpenoids family. Then another property is also having a medicinal property. It is again obtained from eucalyptus. It is having a antimicrobial, analgesic, anti-inflammatory, treatment of bronchitis and all that is there. So what I want to say, this is now the last slide of related to the introduction to the terpenes and terpenoids. Terpenes are very wonderful molecules prepared by the nature. Suppose they are not, suppose for a moment, imagine that they are not prepared by the nature, then what will happen to our life? This curry will not be the delicious as we eat today. And we go for many parties. So at that time we use perfumes and deodorants. So Jack can't go to the party. And Rose will not smell the way it does, and it will be the big problem for the youngsters. And last one and very important is that no rubbers, no tires for vehicle, no balloons and no gloves. So what will happen to all that? It means ultimately that terpenes are indeed very essentials. And as an organic chemist, we should inculcate interest among the students for this particular topic. I'm only discussing here the biosynthesis of terpenes. Introduction part is completed now. Now next one is a biosynthesis, but in between that, structured determination of this compound, which is very much explained in FINAR volume 2, organic chemistry by FINAR volume 2, you may refer it and try to get about the some aspects of the structural determinations. So there are 3 learning objectives as per the terpenes and terpenoids are concerned. One is the introduction, structural features. Second is the structure determination. Third one is a synthesis of these compounds in the laboratory. And fourth one is a biosynthesis. So that makes a complete 1 unit on the terpenes and terpenoids in the curriculum. Now let us talk about the biosynthesis of terpenoids. So this is now whatever the discussion we have made. Now that discussions will be useful to us to understand the biosynthesis of terpenoids. As already mentioned that isoprene is a repeating unit. So first of all, we have to understand the analog of isoprene unit. Isoprene unit is as such not present in the nature. Its analog is present, and therefore we have to understand the genesis of biological isoprene unit. We will see how this 5 carbon atom unit is framed, and we will try to understand the reactivity of this 5 carbon unit so that it will react with the itself or another one so as to get a 10 number of carbon atom. 10 will react with the fresh 5 carbon. It will give 15 number of carbon atom. 15 can react another with the next 5. You can get a 20 number of carbon atom. So it is simply a carbon-carbon bond forming reactions. And the basis of this carbon-carbon bond forming reaction is a carbocation chemistry. It is a purely an organic chemistry, a very preliminary organic chemistry of 10+2 level. Once we understood the biological isoprene, next task is to connect one isoprene with another. That's a condensation. We will get a acyclic analog. And that acyclic analog through enzymatic reactions may undergo cyclization to get a bicyclic, monocyclic, bicyclic, tricyclic, and even a tetracyclic structure like a cholesterol molecules. So this is in a stages we will understand. That is first of all genesis of biological isoprene unit. Now as we know that the nature's laboratory is different. Nature is not having the fine chemicals that are typically present on the shelf of a chemical laboratory. So they are having their own reagents. They are having their precursors. And for that, we should have a background of very simple things. One is enzyme and the coenzymes. Enzymes and the coenzymes are responsible for all the biological processes in the living systems. If you are having a background of that, it's well and good. If it is not at all, you need not to worry. Through this lecture, I'm very sure you will understand it. Now, enzymes are basically a protein molecule. Protein molecule are made up of amino acids. So we are having protein molecule, which is an enzyme. And to this enzyme molecule, a non-protein molecule is present, attached. In a small organic molecule is attached to that enzyme, and it is called as a coenzyme. Now for functioning of the enzyme, we require many times coenzymes. As far as the biosynthesis of terpenoids is concerned, we require the help of 3 coenzymes. One is nicotinamide adenine dinucleotide phosphate, NADP. The word itself indicates there is a nicotine molecule, adenine means nitrogenous bases, dinucleotide means the sugar, and phosphate means is a linkage. We'll have NADPH or NADP plus as a coenzyme which will require. Second enzyme is an adenosine triphosphate, ATP molecule. So adenosine, again we are having a sugar and a nitrogenous base, right? It's a nucleoside and it is connected to the 3 phosphate groups. That's a triphosphate group. And we can have a version of that like a diphosphate or a monophosphate. Now, third enzyme, it is not mentioned here. Sorry, it is mentioned here. It's a coenzyme A. Coenzyme A is mentioned there. Coenzyme A is a very big molecule having SH at the terminal end. We will have some structural features of these 3 enzymes. We will have a shorthand notations of this, shorthand structural notations of these 3. And then we'll talk about their enzymatic reactions. What kind of the reactions? Glycan reduction, glycan condensation. Now, since it is done by the nature, we call it as a biological glycan condensation, or you may call it as a enzymatic glycan condensation. Now, reduction, enzymatic phosphorylation, enzymatic isomerization means it is done by enzyme. Otherwise, glycan condensation, we all are good at that. Reduction, pyrophosphorylation, isomerization. These are very fundamental terms involved in common organic chemistry. So we'll deal with these reactions with the help of these coenzymes. So first of all, let us have an understanding of the first coenzyme, and its name is nicotinamide adenine dinucleotide phosphate, that is NADP+. So here is a big structure, and if you are afraid of that, please ignore it. Only you remember this particular portion highlighted in blue color. We are having a positively charged nitrogen and a nicotinamide moiety. So the shorthand notation, if you consider here, this is a shorthand notation of this molecule. And we are having, this is a NAD+, it gets converted into NADH. We will require NADH in our presentations in the biosynthesis of this molecule. This nicotinamide adenine dinucleotide hydrogen, it acts as a reducing agent. If chemists want to do the reduction, we use sodium borohydride or lithium aluminum hydride. But the nature is not having such kind of the fancy chemical. What they have NADH. NADH is described as a natural sodium borohydride. It's a natural sodium borohydride. This natural sodium borohydride will transfer the hydride to the molecule where it is getting reduced. For example, in this molecule, we are having 2 functional groups likely to be reduced. One is here a carboxylic acid and another one is a thioester. Now, NADH will show a selectivity in the reduction. It will reduce selectively this thioester to corresponding alcohol. Very simple reaction. And that we should understand here because we will require in the biosynthesis of biological isoprene unit. So what I told you that it's a very big molecule that's called as a coenzyme and its name is NADPH or NADH. And it acts as a natural sodium borohydride. It's a hydride transfer reducing agent. And it reduces functional group like thioester and convert into alcohol. So this is the only thing you have to remember. Now let us talk about the next one. This is adenosine triphosphate, that is ATP molecule. Now let us consider a very simple transformation of converting alcohol into corresponding azide, for example. Yes, this slide is very interesting, and this is a structure of adenosine triphosphate ATP. Now, if chemists want to convert alcohol into some substitution reaction, what it does, it reacts, OH is a very bad leaving group, and for that, OH is converted into a good leaving group by treating with the tosyl chloride. So alcohol is transformed into tosyl chloride. Tosyl chloride is reacted with the base with the nucleophile, and we get a nucleophilic substitution. This is a routine organic chemistry strategy to perform functionalizations of OH. But the nature is not doing like that. It does not have a tosyl chloride. What it has a phosphate group, analog of tosylate. So adenosine triphosphate, where there are three phosphoryl group, so it will transfer it to the alcohol. It will transfer it to the alcohol. See here, just we have seen here this OH is converted into OP. 1P stands for 1 phosphorate, phosphoryl group. 2PP stands for diphosphoryl group. So OH is converted into a good living group because of the introductions of phosphoryl group. And once it is a good living group, we will invite a nucleophile. Right? So in this way, you have to only remember that ATP molecule is an analog of tosyl chloride. Tosyl chloride, the role of tosyl chloride is done by the ATP molecule. Right. And the last coenzyme that we require is a coenzyme A. Coenzyme A is a very big structure, and really it creates a lot of burden on the memory. If you try to memorize it, do not memorize it. No need. What you have to only memorize in this structure of coenzyme A is this SH. SH is a terminal end of coenzyme A, and it is called as a business end of coenzyme A. So coenzyme A can be represented as coenzyme CoASH. So typically it is a thiol. Coenzyme A is typically a thiol, and this thiol is acylated by lactic acid through some enzymatic reactions, and it leads to form acetyl coenzyme A. Acetyl coenzyme A is a very important building block in the biosynthesis of terpenoid molecule. What I want to tell you, again I want to repeat, coenzyme A contains SH as the terminal end, and typically you can call it as a thiol. This thiol molecule is acylated by lactic acid, and as a result of that, what you get here is a acetyl coenzyme A. This acetyl coenzyme A is a starting point for biogenesis of terpenoid molecules. Now, if you consider this acetyl coenzyme A, it is simply a thioester, and we know about the esters. If you compare the reactivity of these two, you will find that it is more electrophilic in nature. It is more reactive. See, the nature is very systematic. The nature is very supreme. It is not carrying a reactions with the ordinary ester. It is carrying a reactions with the thioester. The size of sulfur and the carbon atom, there is a substantial difference. Carbon is of small size, sulfur is of big size. So this particular bond is a weaker bond. And that's why here the departure of this thiolate anion is very easy compared to this oxyanion, alkoxyanion. And that's why this thioester are very important reactive molecule. Now with this background, okay, what I have told you in the second half, I told you that this is induced by enzymes. And we require three enzymes, three coenzymes. One is acetyl coenzyme A, second one is a NADPH, and third one is a ATP. These three molecules I have explained you. Now the role of these three molecules I will explain you in this biosynthesis of biological isoprene units. So we are now marching towards the biogenesis of isoprene unit. It is nothing but a carbon-carbon bond forming reactions. Now how it occurs, let us see here. Three molecules of acetyl coenzyme A undergo series of reactions. And what are those series of reactions? I will show you in the next slide. This is a summary of biological isoprene unit genesis. Three acetyl coenzyme A connects together and we get an intermediate called as a mevalonic acid. This mevalonic acid again undergoes series of transformation and we get isopentenyl diphosphate. See here, 5 carbon atoms are here. We started with 3 molecules, 6 carbon atoms will be there. 1 carbon atom will be lost in the form of carbon dioxide through enzymatic reactions. And it is an isopentenyl diphosphate. This is a terminal reactive double bond. In presence of an enzyme called as isomerase, it isomerizes to form dimethyl allyl diphosphate. This is a trisubstrate double bond. Right? So we are having a terminal double bond and we are having a trisubstrate double bond, and moreover we are having allylic here, allylic kind of the system. Allylic substrates are more reactive for nucleophilic substitutions, and we will take the advantage of these two very soon. So what I want to tell you that three molecules of acetyl coenzyme A, they will form 2 biological isoprene through series of reactions. And what are the series of these reactions? Let me explain you here. So this is now a detailed kind of the explanations about the biogenesis of biological isoprene unit. So 1 molecule of acetyl coenzyme A, 1 molecule of acetyl coenzyme A will act as a nucleophile, and the second molecule will act as an electrophile. And these reactions will be carried out by the enzymes. Enzymes contain several basic sites as a side chain. This basic side chain will induce a Claisen condensation. We have learned the Claisen condensation of ethyl acetate to form acetoacetic ester. It is just like acetoacetic ester synthesis, and you may call it as a biological acetoacetic ester. And we have seen how acetoacetic ester is a versatile building block. We are having independent chapter on acetoacetic ester and malonic ester in Morrison and White. So if you solve exercise at the end of chapter from that particular textbooks, you will realize that this is a very versatile building block, and nature is also doing the same chemistry, but it is doing in a very different manner. So first step here is a CC bond formation. We started with 2 carbon atom plus 2 carbon atom, we get a 4 carbon atom. We have to reach to the 5 number of carbon atoms. Then next molecule again comes into picture and it reacts with it. And now aldol condensation occurs. It's not condensation, it's aldol addition reactions occur. Sorry. So this aldol addition will occur. Claisen and aldol will give a beta hydroxy carbonyl compound, alpha and the beta hydroxy. Right. Now, while doing so, there is a simultaneously the hydrolysis of acetyl coenzyme A. Perhaps you may be knowing that there is a lock and key model in enzyme mechanisms. So enzymes are supreme. Nature is supreme. They can do many things very magically than organic chemists. So in this aldol condensation, apart from a CC bond formation, hydrolysis of this thiester group also occurs, and we get this. Now in this case, we are having 6 number of carbon atoms. Right? We do not want sulfur here because our demand is of 5 carbon atom, and we do not want 1 carbon atom. Now for that, here subsequent reactions occurs. NADPH acts as a reducing agent. It reduces this thiester to form corresponding alcohol. We get here alcohol formations. And this alcohol, we are having now how many alcohols? We are having 2 types of the alcohol. One is a tertiary alcohol and a primary alcohol. Both of them are getting pyrophosphorylated. Now during the pyrophosphorylation, what happens? One OH group is converted into diphosphate, and the tertiary is hindered. It will accommodate only 1 phosphate group. This is a very wonderful stage of the reaction mechanism or overall synthetic sequence. Now a stage is reached where this OH is converted into a very good living group, and as soon as it departs, it will also promote decarboxylation reaction. So 1 carbon atom is lost because of this good living group ability of the OH. Good living group ability of the OH is done by ATP molecule. So see here, we have seen the enzymatic reduction by NADPH. We have seen ATP's role here, and we have seen that the acetyl coenzyme A is a precursor for this overall synthesis. And then it is lost, and what we get here is a first biological isoprene unit. It's a terminal double bond. and this way we are getting. Then it isomerizes to form here a trisubstrate double bond. And these two units, IPP, DMAPP, they are called as a biological isoprene units. These are the two molecules, it is not one unit. Why two units are there? Because we have to club together. One isoprene unit will be connected to the another one here. We are having a terminal double bond at this stage, and we are having a trisubstrate double bond. This is nucleophilic in nature. It is electrophilic in nature because of the allylic nature here. We'll see the details about this. So what I want to tell you here, that if you understood it, we can go to the next slide. This is a very important portion as far as the biosynthesis is concerned. We have understood here how the isoprene units are generated. We do not have isoprene molecule as such. We are having an analog of biological. We are having an analog of isoprene unit. They are called as a biological isoprene unit. So we are having an isopentyl diphosphate and dimethyl allyl diphosphate. So these are the two. Now once it is understood, the next here we'll talk about now how this family of the compound is prepared. So we are having here a terminal double bond, and here is a trisubstrate double bond. Let us try to understand the reactivity of these two. This is very nucleophilic in nature. It can attack on electrophile, and as soon as it attacks on electrophile, a tertiary carbocation is generated. And it is this tertiary carbocation which drives the entire terpene chemistry. It is purely an organic chemistry. And then this is an allylic position. This allylic position is prone for nucleophilic substitution. OPP can go and it can attack here. So we are having two different kind of the reactivity with the biological isoprene unit. One is reactive at double bond, and the second one is prone for nucleophilic substitution. So we'll take the advantage of these two. Sometimes they are also called as a hemiterpenes. Right. Now what happens? It acts as an electrophile. This acts as a nucleophile. And it attacks here. We get a tertiary carbocation. See here, it's a very important and a very fundamental step in the biosynthesis of terpenes. We have connected one isoprene unit with another isoprene unit through the carbocation chemistry. Tertiary carbocation is generated. That's why head to tail linkage is there. See, these two can react just to get a tertiary carbocation. See, this is a tail, this is a head. Why there is a head to tail? Now here you will find a reason. Reason is that tertiary carbocation is more stable than anything else, and that's why we get this species. Once you get a tertiary carbocation, now rest of the portion is very simple. Hydrogen will be lost and we will get a monoterpene molecule. It is called as a geranyl pyrophosphate. There may be thousands of monoterpenoid molecule containing the 10 number of carbon atom. Their biogenetic precursor will be this molecule only. That you have to understand. Right? Then this geranyl pyrophosphate clubs with the next molecule. See, this is again allylic. This is a terminal double bond. It's a allylic terminal double bond. So this, this is getting repeated. The first step is getting repeated at the second one here to get a here cis-Q terpenoids. This is a reactive double bond. It's an allylic reactive position. So again, new tertiary carbocation will be generated. This will again lose a proton here, and it is called as a farnesyl pyrophosphate. This farnesyl pyrophosphate is a precursor for entire family of cis-Q terpenes. Whatever the 15 number of carbon atoms are there is due to this one. Now again we can add one more. We started with C5 plus C5, you get a C10. C10 plus C5, you get a C15. C15 plus C5, you get a C20. And this way we are addressing to all classes of terpenoids. Here again a tertiary carbocation is generated, and you will get a geranyl pyrophosphate. We will have like this, right? Now apart from this cascade of reaction, that is adding one terpene to the another, one isoprene to the another, there is a second possibility. And what is the second possibility is that this farnesyl pyrophosphate, there's a C15 and the C20, they can undergo dimerization. And that dimerization will give you triterpenite. Dimerization of diterpenite will give you tetraterpenite. So because of this dimerization of that, see, C40 will not be obtained like C5, 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 C5 up to C20, it will not occur like that. Or C30 also, it will not occur like that. C30 is likely to occur by combining with itself. C50. 20 number of carbon atom can combine with itself, we can get a 40 number of carbon. And because of that, at the central portion of that molecule, there's a tail-to-tail coupling. We have seen in the beginning, exceptions occurs to the isoprene rule. Head-to-tail is a very abundant. But at the central portions of triterpenite as well as a tetraterpenite, we are having a tail-to-tail linkages. It is only because of this particular reactions involved, right? So this way I have explained you. This way I have explained you how this all classes of compounds are generated. We started with C5 added to C5 head-to-tail, C10 added to C5 C15, C15 plus 5 C20. You can get a C25 also, but C25 examples are very less in the literature. Alternately, C15 can combine with itself, you get a C30. C20 can combine with itself, you can get a C40. C30 is a precursor for entire steroid molecule. Cholesterol is having 27 number of carbon atoms. And again, it's a very interesting class of reactions about the biosynthesis of steroids. In all these cases, we are having head-to-tail linkages because tertiary carbocation is intermediate there. Here, tail-to-tail linkages is occurring because there is a dimerization process. Now, after this, we'll talk about an individual class of terpenes. That's a monoterpenite, diterpenite, triterpenite, and we'll talk about why do you get a tail-to-tail coupling, and then our lecture will be completed. Okay, so we are having now here this particular biosynthesis of monoterpenes. Now we will see how they are obtained. We'll have some examples here. We get geranyl pyrophosphate, I have already explained you. And once we get a geranyl pyrophosphate, from that you can get a geraniol. You can get a geraniol is present in rose oil, myrcene is present in a bay leaf, linalool is present in a coriander. Okay, so see how it is prepared. OPP is a very good living group. It can generate a carbocation. This carbocation can be attacked by water. We'll get a geraniol. Once we get this carbocation, it will undergo resonance. We'll get a tertiary carbocation. This tertiary carbocation can lose a proton. We'll get a myrcene. This tertiary carbocation will be attacked by water. We'll get a linalool. So see, all these three molecules are prepared from geranyl pyrophosphate. And how do you get a geranyl pyrophosphate? We get it from dimethyl allyl pyrophosphate and isopentenyl pyrophosphate. We were having two precursors called as a inhibitor. Okay, then let us see how again limonene is obtained. We are having here is a double bond. This double bond can undergo isomerization through an enzyme called as isomerase. We can have a loss of this pyrophosphate group, carbocation will be there. This carbocation will be trapped by the double bond. We get a new carbocation. Now this carbocation will be trapped by water. We'll get a alpha terpenol, which is present in the cardamom. Then this proton can be lost. We'll get a limonene. Limonene is present in a sweet lemon. That's orange. That's a Mosambi. And it can also lose this one. It can get a terpenoline. Okay. Again, see here, we have seen already the monoterpenoids. Let us see the bicyclic terpenes, how they are generated. Now this carbocation this carbocation can be trapped intramolecularly by the double bond. There are two positions. It can go to the 5 or it can go to the 6. If it goes to the 5, you get a new carbocation. This carbocation can lose a proton. You will get alpha pinene and the beta pinene. These two are generated. Similarly, it can connect to the 6 number carbon atom and we get a camphor. With the help of camphor, we pray to the God every day. So camphor is also possible to get it. And what is a precursor? Precursor is geranyl pyrophosphate. And how do you get a geranyl pyrophosphate? That I have explained you already. Right. Then again, it is a summary of all these discussions what I have made you. This is a geranyl pyrophosphate and we have prepared now how many molecules? 1, 2, 3, 4, 5, 6, 7, 8, 9 molecules as a representative class of this. It's a very preliminary organic chemistry and the students should be encouraged to prepare this kind of the class, such type of the topic, because they will be fascinated by the nature. It's a nature's chemistry. Nature is doing this kind of the reactions through enzymes. Oxygenated form, that's a reaction with water is there. And why it is common? Because tertiary carbocation is a intermediate there and it will be definitely trapped by the water and that. So this way, we are having now some illustrative examples. This is again a chart, very exhaustive chart. It will not be possible to write all these things. Geranyl pyrophosphate gives you this carbocation. And from this carbocation, you can draw a, organic reaction mechanisms for biosynthesis of all these molecules. I think out of this chart, 9 or 10 molecules I have already explained you. Alpha terpenol, limonene, alpha terpenol, terpenoline that I have explained you. Then camphor, alpha pinene, beta pinene that I already explained. Campene is also possible to write very easily. This is also easy to write. So this way, students can undertake the mechanism of these reactions in the examinations. Now, we have completed the biosynthesis of this compound. Let us talk now about the cis-Q terpenoids. In cis-Q terpenide, farnesyl pyrophosphate is a precursor. This will be lost, we get a carbocation. Carbocation will be trapped by water, we get a farnesol. Right. Then again, this carbocation can undergo resonance and we can have a loss of proton. Its name is farnesyl. Then alpha bisabulin, gamma bisabulin, and the beta bisabulin. This is again a nomenclature. Beta stands for trisubstrate double bond, and the gamma stands for here. Gamma stands for here is exocyclic double bond. I think these 2 points are common. It is only differing on this double bond. Alpha, beta, and the gamma are related to the nomenclature and the nature of substitution pattern on this particular double bonds. So beta bisabulin and the gamma bisabulin. This is all about the cis-Q terpenes. And again, similarly, we can have here all range of this cis-Q terpenide molecules. So if you are teaching at UG level, small molecules will be sufficient. But if you are teaching at the PG level, then you can give such a complex molecule for writing the structure, writing the reaction mechanism to the students. Yes. So it is there. I will share this slide with Professor Podape madam, and you will get the access to all these contents, and it may be helpful to you perhaps. At that time, you can undertake some writing of the reaction mechanisms. Yes. Now, the last point, and a very important, tail-to-tail linkages. Head-to-tail linkages was possible because tertiary carbocation is the only cause. But tail-to-tail linkages, here it is also occurring. And what is the reason, how this is occurring in the nature, that I want to explain you here. So C15 will club with another C15 molecule, leading to form C30 molecule. In this C30 molecule, that is a squalene molecule, we will find head to tail linkages everywhere. Except at the center portion, there is a tail to tail linkage. Now, how do you get this tail to tail linkage? For that, this reaction pathway is written. You will find its more detailed discussions in the clade. Now, this is a normal reaction. One act as a electrophile, another act as a nucleophile. See, in the earlier case, there was a terminal double bond and there was an internal double bond. Here, there is nothing like that. This is a reactive allylic position, reacts with this double bond, and we get a tertiary carbocation. It's a normal CC bond formation. Now, this carbocation undergo a very different reaction, and for that, organic chemists do not have any explanation. The Claydon, the author says that only enzymes can do this remarkable reaction. Here, hydride, here this hydrogen is removed. It's not acidic in nature, in fact. It is removed by some enzymatic site, and you get a carbanion. This carbanion cyclizes here, you get a cyclopropane. This CC bond formation is a remarkable reaction carried out by the enzymes. No ordinary chemist can do it. And once we get a cyclopropyl ring, it undergoes neighboring group participation. That is a relief of strain, and you get a new tertiary carbocation. This tertiary carbocation is stabilized by ring opening here to form a allylic carbocation. And this allylic carbocation will be treated with or will be allowed with the NADPH. It's a hydride one. It will react with the hydride source through NADPH, and we get a squalene one. So this is very interesting. Here the only point is about this cyclopropane formation, which is done by the enzymes because of the lock and key model. And that's why you get a squalene one. Right. So this way I have also explained you tail to tail linkage. Now, we made a lot of discussion. The point is that what is the evidence in favor of this biosynthesis? What is the evidence? Evidence is done by isotope labeling experiment. Chemists have injected isotopically labeled acetyl coenzyme A into the plant. And as a result of that, they isolated at the end the product. Support of the proposed biosynthetic pathway. Number one evidence. Second, mevalinic acid. Let us complete this discussion. Again, even in a squalene molecule, see, this is observed alternately. But here there is a tail to tail linkage. These two are in the rest of the positions, there is an alternate label, but at the central there is a tail to tail. This kind of the isotopic distribution was observed, and that's why the proposed biosynthetic pathway is accepted. And the second one is a mevalinic acid. I have shown you somewhere mevalinic acid as this is a mevalinic acid. This mevalinic acid is isolated from the plant many a times. And since it is isolated, it means that it must be involved somewhere in the biosynthesis of this terpenoid. Now, let us talk about the last portion. It is not involved in our lecture, but how much time is left? So I will cover this. Now, the next category is the steroid molecules. Steroids are tetracyclic molecules, and they gives you this hydrocarbon. That's the definitions and all that. We will not go into that. Now, how cholesterol is prepared, that I want to show you here. This is a squalene molecule. Squalene molecule will undergo epoxidations at the terminal end through an enzyme called as a squalene epoxidase. And once we get this epoxide, it undergo cascade of reaction. It's a very joyful to write one reactions at a time. But here I have shown you the cascade of reactions. And this cascade of reactions will give you a tetracyclic structure responsible for cholesterol. and then it undergoes some series of reactions to get a lenosterol. Biosynthesis of lenosterol should be taught to the students. And this lenosterol undergoes subsequent 19 steps to get a cholesterol. That's beyond the scope of our discussion. But the lenosterol, how do you get it? By a cascade of reactions. Now, in this case, how epoxidation occurs? Epoxidation occurs by Fa, flavonine, adenine, dinucleotide, dihydrogen. Okay. So this FAD reacts with the oxygen. It forms a peroxide linkage. This peroxide forms an epoxide. For carrying out epoxidation, organic chemists use MCPBA, metachloroperbenzoic acid, or a perbenzoic acid. But here the nature is using molecular oxygen. Molecular oxygen is treated with FADS2. It forms a peroxide, and this peroxide transfers oxygens. And it is a very crucial step of biosynthesis of lenosterol. So this way it happens. You can see here also epoxidation, and then one by one the rings are preparing here. So this will form, we'll get first ring, second ring, third ring, and the fourth. Okay, so this topic is not just way of looking at my PowerPoint presentation or reading the textbooks. It is necessary that we should take a pen and paper and let us make an adventure of writing such kind of the reaction sequence. Perhaps it will be the more joyful journey if we undertake such kind of the things. Okay, so here I would like to stop. I have again the next topic is alkalite that I do not want to cover here. I think I have taught you almost 1 hour 5 minutes. Coordinator, madam, you are there? So if you are having any difficulty, any question, it will be my pleasure to answer all the questions, the discussions. So this is a very beautiful slide that I want to show you. I had been to the Ooty, and this is a plant. Where the eucalyptus was distilled off. So this is a all kind of the oils. Its base is terpenes chemistry. Okay, and if you go to the Ooty, try to take some chemicals out of that. They are really very pure and very peaceful. Okay, so thank you very much for passionate listening. Now the session is open for the discussion.
Hello.
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 Sir, phenolic monoterpenoid can possible synthesis by this method?
Can you repeat the question, sir, please?
Phenolic monoterpenoids.
Phenolic monoterpenoids. For example, tocopherol.
Okay.
Tocopherol contains the phenolic moiety, but the side chain is present in a terpenite form. Means there's a, I think, phytol ring chain is there in a tocopherol.
But by this biomethod is possible, sir, synthesis? Phenolic
Yes,
compounds?
Biosynthesis, nature has done it already. It means biosynthesis there. If you go to the more particular literature of this, you can get more insights on that.
Okay. Thank you, sir.
Any question?
In your biosynthetic procedure of these terpenoids, that monoterpenoids, SQ-terpenoid, triterpenoid, which factors control the chain length? Is it exclusively dependent on the pocket size of the enzyme? Because there is a possibility of elongating the chain length, for example, from C30 to C60 or C45. So my question is that which factors exclusively controls the chain length? Is it only the pocket size of the enzyme? That means the reaction is exclusively enzyme dependent?
Yes. Actually, the very important slide. Hello. The very important slide that I showed.
Yes, second year. Yes.
This is geranyl pyrophosphate. Once it is obtained, rest of the thing is an enzyme chemistry. I have shown you somewhere isomerization, resonance, rearrangement, trapping of carbocation by the double bond. So
That means
plethora of structures are possible.
yeah, but the controlling the chain length, is it enzyme dependent? That means
Actually, you are talking about that steroid synthesis.
no. This one, this biosynthesis of terpenes. You look here, you have synthesized C10, C15, triterpenoids C30, tetraterpenoids C40, but there is every possibility of obtaining C60 or C45 or other chain lengths.
Actually, this pattern C25 or C55 is generally not observed in the nature.
Okay, so
They are not observed in the nature. Perhaps maybe again lock and key model may be involved there.
that means it is enzyme dependent process.
Exactly, enzyme. It's an entire enzyme chemistry.
Yeah, that means, so is it possible to have a biomimicry of this synthesis, this particular terpene biosynthesis? Is there any report?
What you are talking, actually your voice is not getting clear.
Is it possible to have possible to do a biomimicry of this process? That means can it be done in the laboratory?
Oh,
controlling the chain length?
No, you can't do this one in laboratory. That's why it is called as a biosynthesis. You know, the nature is a laboratory. The living cell is a laboratory. You cannot do in a flask.
Okay. And one more question is that here the carbocation. These carbocations have many possibilities. That can be trapped by water molecule, or that can also be trapped by OPP minus. Because TPO minus is also a reasonably good nucleophile, but it undergoes an elimination reaction. You have shown in another slide that this mechanism is somewhat is going through a tricyclic carbocation intermediate that is converting to tetracyclic carbocation intermediate, right? The mechanism is also applicable here in this biosynthetic process.
And then the trapping of carbocation by OPP is not possible. Otherwise, this is a tentative pathway adopted by the chemist to understand the structures of wide range of terpenes and terpenoids. You know, more than 23,000 terpenes and terpenoids are known. So addressing the biosynthesis of each molecule will be extremely difficult. But this general presentations accounts for the biosynthesis of large molecules.
Okay.
Sir, what is the specialty that this acetyl coenzyme A acts as a precursor for maximum pathways? Sir, what is the specialty of acetyl coenzyme A that nature has selected this very molecule?
Acetyl coenzyme A is a thioester. Thioester is not a normal ester. It's a more reactive ester than the normal ester. And that's why the nature is utilizing that. Thioester is having enhanced reactivity compared to the ordinary ester, and that's why we say that the nature is supreme. Nature is having extraordinary reagents with them. Acetyl coenzyme A is a thioester, and thioester, or forget about the ester, but we know the chemistry of the ester. So that way it is utilizing here in the CC bond formation. Yes, any more question? Very good questions are coming. Really, I am very happy.
Sir, I want to ask one question, maybe little off the beat in the sense, some natural products, have you isolated the natural products or just this is the synthesis?
There is a protocol for isolations of many natural products available. You can go through Google also, or you can go through any literature also. It's a very common one. It's not a big thing.
Yeah. So sir, then my question is like, you know, one of the medicine which is ashwagandha used in Ayurvedic medicine. So to find the active ingredient from that, what method would be the, the most appropriate method?
Actually, you have to go to the literature survey. You have to find out that whether somebody has done work on ashwagandha earlier or not. Number one. Number two, then you trial and error. You make a fractionations by making combinations of polar and non-polar solvents. then every time you take a TLC and then try to find out the chromatography, and after chromatography you will get a product in a pure quantity, send it for NMR and the mass, and then you reach to the structure. This is a very laborious work to be carried out, and it requires a lot of sophistication in the laboratory.
Right.
Many times it is done by HPL cells nowadays.
Right. Okay. Thank you, sir.
Any other question? I hope that you make this part, you make a part of your curriculum, this particular topic. Natural product chemistry is the original organic chemistry. You know, people teach substitution reaction, elimination reactions, addition reactions. And when it comes to the natural product, they say that it's no kuch bhi nahi hai. But I think it's a very rich chemistry. It's a 10+2 level chemistry. And you convert that concepts. We have to apply the concept to the nature. So you make it more interesting by incorporating this particular topic and try to have problem-solving approach. In Morrison and Boyd, at the end of chapter, many problems are given on terpenes and terpenoids, where the ozonolysis experiment is given, where the degradation experiments are given. So you make a list of all the problems and try to incorporate it. Any other question, please?
Good morning, sir.
Good morning.
Much thanks to you, sir, for such a detailed explanation of the topic.
Thank you.
I just wanted to know, can you suggest some books for PG level of course for this terpenes and terpenoids for such a detailed and thorough understanding of the topic? Like IL Finar is there, but some other suggestions from your side, like for PG students?
Actually, I searched many books on natural product chemistry, but I found IL Finar Volume 2 as a supreme book for this particular topic.
Okay, like sir, for nomenclature and those introductory parts, is there any like some other book also?
In my opinion, you read twice, thrice IL Finar, there itself you will find many things. Perhaps one round of reading may not get, because the printing pattern is not a modern one. It's a very old one. That's why sometimes we don't read it very carefully. But IL Finar Volume 2 is the best book for that. And many Indian authors have make a notes of IL Finar. You can refer that also. But the original material is you'll find in IL Finar Volume 2. Yes, is it okay?
Yes, okay, sir. Thank you.
I'm very happy to see that many students are from Nagpur University for this refresher course. Yes. Any other question?
Yes, sir. Very good morning, sir.
Good morning.
Sir, since you have taken the pyrophosphate pathway, did you apply any gem dialkyl rule to predict the stability of the cyclic terpenoids?
Geminal dialkyl. Not understood your question.
Gem dialkyl rule, which is used to find out the stability of monocyclic or bicyclic or tricyclic.
Yes. It is there. That substitution pattern for bicyclic, there are various possibilities, and out of that, what you are talking about, the geminal dialkyl.
Yeah.
So it is definitely there. It is given in the beginning, perhaps.
Okay. Since you have shown many acyclic compounds, did you do any cyclic compounds, synthesizing cyclic compounds method by using pyrophosphate pathway?
Can you type your question, sir, if you don't mind? I will understand it better.
Okay, sir, I will type it.
Any other question, please?
Sir, I have a question. Hello.
Yes, please.
Yeah, my name is Sarat Babu. I am working as assistant professor in Kalki College, Delhi University. So my question is that why all plant or flowers are synthesize the terpenoids or alkaloids? Why the particular compounds? Each flower has a different terpenoids. Each fruit has some.
Your voice is not audible, please. Can you speak loudly or can you give me the message?
Yeah. Now it's audible?
Now just little audible than previous one.
Okay. My question is one thing. So why the plant or flowers synthesize the terpenoids?
Yes.
Particular compound. It's say every plant has a different terpenoids. Why the plant want to synthesize? That's
Yes. That's a great question you have asked. The purpose of synthesizing this terpenoid molecule is for three different functions. One is a communication, another one is a defense mechanism, and the third one is there that I forgot, but it is for defense mechanism many times. The plants get wound, and to recover that wound, many times the resin is generated. That resin is nothing but the bioterpene. That is nothing but the terpene. So that is a very important function. The third function is attraction, to attract this butterfly and all that. So for three purposes the terpenes are useful. One is for communication purpose, another is for defense mechanism, and third one is for attracting the insects.
Okay, pollination.
And corresponding to these three purposes, there are very different examples given in the literature also. These examples are not on my fingertips, unfortunately.
Okay. Thank you. Thank you.
Stability of cyclic compounds.
Any other question, please?
Sir, this same unit is used in the rubber. Isoprene unit, I think, sir, in the rubber, it is also used to make rubber.
Yes, your voice is again not.
Sir, this isoprene unit, sir, it is also used in the rubber as a unit, as a monomer. When we make polymer,
Yes,
same isoprene unit is there.
Yes, natural rubber is a polyisoprene molecule. It's a rubber, and rubber means it's a gloves, tires. I have already mentioned in the beginning. Any other question?
Sir, all these molecules are pharmacologically active.
Yes, there are many molecules pharmacologically active. I have shown you one example of Taxol, which acts as an anti-cancer one. And eucalyptus oil is having is used for bronchitis, for treating the bronchitis. Yes, there are many medicinal applications of this also. Any other question? I think there is no question. Am I right?
Yes, sir.
Hello, sir. My voice is audible? Yes or no?
Yes, it is.
Yes, sir.
Yes, please.
Okay, sir. So, myself, Dr. Bhavesh Bharatiya. I am from Sardar Patel University, Vallabh.
Oh, great.
Okay, sir. So, if no question, so can we conclude the session?
Yes, please conclude the session.
Okay, sir. So, good afternoon, everyone. It is my absolute privilege and honor to propose vote of thanks on behalf of UGC MMTTC Center of Sant Gadge Baba Amravati University and all the participants of this refresher course. I extend our deepest gratitude to our esteemed resource person, Dr. Nand Kishore Karanade sir, professor from the Department of Chemistry, Nagpur University. Sir, your expert session on the biosynthesis of terpenes and terpenoids that leading to the natural products was incredibly illuminating and intellectually stimulating. Sir, you broke down the complex biochemical pathway into highly accessible concept. Your deep expertise gave us fresh, valuable insight to bring back to our own classroom as well as research lab. We also expressed the sincere appreciation to the Director of Course Coordinator of Sant Gadge Baba Amravati University and the entire team of the UGC MMTTC. Thank you for seamlessly organizing this session and providing this excellent platform for the professional development. A big thank you to the all fellow of the participants for your active engagement throughout the provoking question during the discussion session. Once again, thank you, Dr. Karade sir, for sharing your time and profound knowledge with us today. Thank you all and have a wonderful day ahead. Thank you, sir.
Thank you. Thank you. I will leave the meeting with your kind permission.
Thank you, sir. Thank you, sir.
Hello, Professor Manjre sir. Hello, Professor Sudesh sir.
Sir.
Sir, good
Yes,
afternoon.
I'm there.
Yeah, sir. Good afternoon. You are very much audible, sir. Shall we start if you could permit, sir?
Yes, please.
Yeah. So, friends, good afternoon once again. We'll moving for the next session. So, first of all, I extend my warm welcome to Professor Sudesh Mandre, sir, to this refresher course and particular to this session. And now may I request Dr. Bhavna Khobragade, madam, to please introduce our guest first, and then we will start with the session. Dr. Bhavna Khobragade, madam, please.
Yes, sir. I'm audible.
Yeah, ma'am. Clear.
Okay. Thank you, sir. Very warm welcome to coordinator of this program, all the participants and technical team to these sessions on topic chalcogen containing molecules to cancer detection and introduction to the isolobal analogy. Of this online refresher course in chemistry organized by the Malviya Mission Teacher Training Center, Santa Gadge Baba, Amravati University. I am Dr. Bhavna Khobragade, Assistant Professor, Department of Chemistry, RDIK and NKD College, Badnera, Amravati, Maharashtra. It is my distinct honor and privilege to introduce our eminent expert resource person for today's session, a highly accomplished researcher and the award-winning academician and the leading global voice in inorganic and the organochalcogen chemistry, Dr. Sudesh Manjre, sir. Welcome you, sir. For our university, today is the proud homecoming moment. Dr. Manjre, sir, completed his bachelor degree from the Vidya Bharati College, affiliated with the Santa Gadge Baba Amravati University. From these foundational roots in Amravati, his academic journey has been nothing short of the exemplary. Dr. Manjre sir went on to pursue his MSc in inorganic chemistry from TC College, Baramati University of Pune. He then cracked the prestigious CSIR, GRF, and the SRF national examinations to pursue his PhD at the premier Indian Institute of Technology, Bombay. Under the guidance of the renowned Professor S.B. Singh, sir, specializing in studies in the noble chalcogen compound. He was awarded the coveted Institute for the Basic Science Research Fellowships to conduct his postdoctorate research at the world-renowned Korea Advanced Institute of Science and Technology in South Korea, working along with Professor David. Currently, Dr. Manjre sir is serving as an assistant professor in the prestigious Department of Chemistry at the University of Mumbai. His core research interests lie at the absolute cutting edge of the chemical biology and material science, focusing on the development of the chalcogen-based molecule for the cancer detection, advanced material for energy conservation, and design innovative molecule probes for the selective sensitive detection of the reactive oxygen species, bithols, and the toxic metal ions to aid the diagnosing disease like the Alzheimer's and cancer. Dr. Manjre sir scientifically carried profound global weight. He held a highly valued Korean patent for the synthesis of the chalcogen-based molecule probes. He has authored 45 highly impact research publications in top-layer international journals like the Dalton Transactions, SET Omega, SES Omega, etc. His scholarly impact is reflected in his staggering metrics, boasting over the 1,378 citations, an H index of 18, and I-10 index of the 25, reflecting his domain mastery. He also serves as the associate editor of the international journal Main Group Chemistry. His excellence has been consistently recognized at the national and international forums. He was the best owned He's the prestigious Outstanding Researcher Awards by the University of Mumbai. He has successfully secured and exploited significant research grant from the apex bodies like the SERB, DSP, Government of India, and UGC. He has dedicated mentor who is successfully guided 5 PhD scholars who are now working in the top industries and pursuing postdocs in the USA and Germany, and is currently guiding 4 more doctorate candidates. He is the short after the speaker globally, having recently delivered a keynote address at the 13th International Symposium and the Selenium in the Biology and Medicine in South Korea.
Ma'am, I think that's it.
Just a
Thank
minute,
you
just
very
a minute,
much.
sir. Just a minute, sir. Your
Yeah.
biodata is just so lonely and very much interesting, sir. Just a minute. Dear participants, we are incredibly fortunate to have the absolute expert who blend deep fundamentals in organic chemistry with the real world biomedical applications. I am certain the sessions will provide immense intellectual value to all of us. Without further ado, I request all the participants to welcome him. Sir, we welcome you. The virtual platform is all yours. We took the forward at the enlightening sessions. Thank you, sir. Welcome you, sir.
Namaskar, sir.
Thank you.
So sorry, sir, I was in lecture.
Yeah.
Welcome you, sir.
Thank you, ma'am. Thank you.
So please, you can. I hand over this session to you, sir. Please.
Okay, ma'am. Thank you, Dr. Bhavna, for a nice introduction. Actually, this much long introduction was not needed. But thank you very much for your kind words. And let me thank you, Amravati University, especially MTTC, for inviting me for this particular refresher course. And special thanks goes to the coordinator of this course, Professor Kodape Madam. Madam always invite me for different kinds of work. So thank you very much, ma'am, again for inviting. I'll share the screen. Just let me know whether it is visible for you people.
Yes, it is visible.
Can you see the
Yes, yes.
Yes, sir.
Slides?
Visible, sir.
And am I audible clearly?
Yes, yes, you're clearly audible.
Okay. So thank you once again. I'm Dr. Sudesh Manjre, as Dr. Bhavna mentioned. I will just, this is our topic for today's discussion, I will say, rather than the presentation. That is chalcogen containing molecules for cancer detection. And at the end, we will go to the introduction to isolobal analogy. So this is Mumbai University campus. Since it is an online refresher course, so I assume that there are many participants from the distinct places, and some may have visited Mumbai University, some may not. So this is a main campus which is in downtown and located near to the Gateway of India, that you can say even the Mantralaya. And then there is another campus, which 2 photographs I have kept at the down, that those are from which we called as Kalina campus, which is situated in Santa Cruz East, near to the international airport. So this is the campus where most of the, we can say, almost all departments are accommodated in this Kalina campus. And Department of Chemistry is also situated at this in this particular campus. Apart from these 2 campuses, we are having few more campuses, like campus, sub-campus in Thane, sub-campus in Kalyan, and 1 sub-campus in Ratnagiri. So this is about Mumbai University. As ma'am mentioned that I'm working in the inorganic chemistry. So specifically I work on the main group chemistry, and in that also we work on the chalcogen group, that is oxygen, sulfur, selenium, tellurium and polonium. Here, as we know that recently or today, the cancer, a major problem for world. And according to who? That is WHO, cancer is leading cause of death worldwide, and nearly 10 million deaths occurs in 2020. And at the same time, new cases in 2020, this is data from 2020, that nearly 2.2 million, the breast cancer, then lung cancer, colon and rectum, prostate cancer, stomach cancers, and so on. So we can see this number is increasing year by year. And we can see that just 5 to 10% is due to the genetic effect, and almost 90 to 95% is due to the environment and lifestyle. Now, since we are chemists, that why this particular cancer happen, what are the species responsible for that? So there are two chemical review paper. These are the review articles by KP Cape in 2012 and by J. Shi in 2019. And what they observed that this amylate cascades, metal ions and reactive oxygen species specifically responsible for the kind of abnormal growth of these cells. And if you see in detail that reactive oxygen species like superoxide, hypochlorite, H2O2, or even the radical, even the RNS, nitrogen species, amylate cascades like biothiols, as well as the amino acids and metal ions. We can see are different metal ions, they are responsible, but they are very much essential for the promote of growth if they are present in the normal level. They regulates the hormones and human metabolism. But if their level is abnormal, then they create oxidative stress, and ultimately it leads to cancer, neurodegenerative diseases like Alzheimer, Parkinson, and the cardiovascular disease. So the detection of these bioanalytes, it becomes an important task, that where they are in large excess, and whenever they are in large excess at a particular place, it means there is something abnormal things are going on. So we use them for the detection of cancer. So for this, we have to have some strategy. And since I'm working on chalcogen based molecules, so we have a strategy that we can have a one that's E means selenium or tellurium. We can have one selenium or tellurium in the molecule, or two, or we can have a dimer of this, or we can have a coordination kind of sites. But this will be a reaction center. Now, reaction of these analytes like reactive oxygen species, metal ions, or what we call the biothiols, they can react with these chalcogens, but the response should give by some other species. So that is why we have embedded here the fluorophore, and we are using a fluorescence spectroscopy to detect the responses. So we can either it can get oxidized with the ROS RNS, we can get a response by changing electronic structure of the complete molecule. Sometimes both if there are two chalcogen atoms present in a dimeric form, then both the chalcogen atoms may get oxidized. Sometimes it will break down and with the biothiols, and it may form like a SCS kind of bond or TES kind of bond, and we will get a response. And if you have a kind of coordinating core, the two seleniums or sometimes nitrogens with them or oxygens, then they can coordinate to the metal ion, and ultimately electronic structure will change and it will give a response. So this is a design strategy for these molecules. And if you see the mechanism that why this particular fluorophore is giving response, so we can understand from this particular diagram that if your receptor means selenium or tellurium molecule, so which is in a generally in a higher energetic than the fluorophore. So when you excite the fluorophore, that the electron goes, once it goes into the excited state, immediately your receptor, that is selenium or tellurium, they will transfer the electrons and you will not get fluorescence. So most of the time this compound, they are in a quenching state. So there is no fluorescence to this particular molecule. But when they react with the analytes, like suppose it reacts with the ROS, so your selenium or tellurium, these chalcogens will get oxidized. Now they are lower in energy. So once they oxidize, that when you excite this molecule, now this will not be able to transfer the electron, and that is why the excited electron can come back again by giving the fluorescence or phosphorescence. So this is the mechanism with the turn off and turn on sensing. So with these things in mind, we have designed particular molecule with the 2-BODP. This BODP stands for boron dipyromethane molecule, and this is highly fluorescent molecule, this which is shown in the green. So this is the BODP molecule. So we have embedded this BODP in particular with the selenium. So you can see the synthetic strategy, which is very easy, and this is a kind of general, a lot of general reactions are known for this synthesis. So first, these methyl molecules converted into aldehyde molecules, and then amine into the bromine. And this aldehyde we can easily convert into the BODP by using the pyrrole and the oxidizing agent in presence of TFA and like BF3 thread. So this is first we have connected with the selenium. You can see that SCPH is connected, and then with the in presence of pyrrole and TFA, this is converted into the dipyromethane. We call it as a dipyromethane because this particular carbon, which is sp3 hybridized, this particular carbon sp3 hybridized. And then we can convert, we can oxidize this by using DDQ and other reagents, which will embedded again BF2 also into the molecule, and this becomes a dipyromethane, and that is why it's a difluoroboron dipyromethane molecule discard. So we synthesize this particular molecule. Since this is the first example, so I will show you some characterization techniques. What are the characterization techniques we have used? So we use first the proton NMR. You can see a very nice peak for the dipyromethane molecule. These 3 protons from this particular pyrrole group, and we have CH, which is present here. And then definitely a selenium NMR, it gives a nice selenium NMR at 276, which confirms the presence of selenium. When we convert it into the BODP molecule, that is dipyromethane, the particular CH that is absent now, there is no CH present, and this pyrrole proton, they have shifted, downfield shift into that. And again, at the same time, selenium gives a downfield shift, so which confirms the formation of this particular molecule, BODP molecule. Now it's time to go for screening, that whether this molecule detects something or it gives a fluorescence or it doesn't give the fluorescence and all that. So we did the photophysical study, and you can see that this particular molecule gives absorbance in the UV, that is lambda max around 500. And when we excite at this particular, it gives a very small fluorescence around 525. Okay. And we screen this molecule with different ROS. You can see superoxide H2O2, then tertiary butyl hydrogen peroxide, hypochlorite, OH radical, then butoxide radical. So this we have generated and we have screened this particular compound with this, and what we observed that this particularly is giving very high response only in case of the superoxide. You can see the other molecule, with the other molecule, there is fluorescence lies with the probe only. that the original molecule. So there is no response for any other ROS. Only for superoxide, when you add superoxide, it gives a response. Now, once you came to know that this is giving response to the superoxide, we try to find out the detection limit for that, and for that we increase the concentration from lower concentration to higher concentration of the superoxide, and from that we came to know that detection limit is 4.4 micromolar. At the same time, it is very important that whatever fluorescence we are getting due to this particular superoxide, it should be stable enough for longer time, so that we will be able to use it in real time detection. And that is why we did the kinetic study. Here we can see that it gives response within first hour, almost 30 minutes, within first 30 minutes. And then it is stable for longer time, almost for 12 hours it is stable. So it is compound is photostable. There is no photo bleaching of the compound, and we can use it. So now why it giving a fluorescence after reaction with the KO2? So whatever strategy we had designed, is it working? So we have checked its mechanism, and here we can see this is a photo-induced electron transfer mechanism. So when the selenium is in the reduced state, we can see that the selenium can donate electron to the body peak core, and the fluorescence is quenched. But when it reacts with KO2 and this selenium gets oxidized, so selenium itself becomes electron deficient. Now it cannot transfer this electron to the body peak core, and that is why after excitation it emits the fluorescence. So this particular molecule we confirm with the NMR also, this proton NMR, and even the mass we can see that SiO2 is present in the mass. So we got M minus H plus P. So after confirming this mechanism, the few more things also we did the I have not shown here actually, that we did the interference study, that in presence of other reactive oxygen species, this is detecting specifically to the KO2, not to the other reactive oxygen species. At the same time, we did pH study, that yes, it is working in the physiological pH, so that we can use for the detection of species inside the cell. So here is the biological study, that this water molecule we have synthesized, yes, it is detecting superoxide, but this should not be toxic to the cell. If it is toxic, then it will not work inside the cell. And that is why we can see that we have checked the toxicity of this particular molecule, and it shows cell viability above 90%. So almost no toxicity of this particular. And then we have checked this compound inside the cell. Since we got the result, whatever photophysical result in the cuvette, whether these are working inside the cell or not, our compound is kind of able to detect this reactive oxygen species inside the cell or not, and that is why we use inside the cell. So we first check the cell, so there is no fluorescence to the cell, you can see here in the diagram. And we then add PMA, that is, these cells were incubated with the PMA. PMA is a natural product which can induce this reactive oxygen species inside the cell. So we check this after incubation with the PMA. Again, there is no response. But when you incubate these cells with PMA and then you add your probe, yes, we can see that it is clearly the cells are clearly visible. You can see that fluorescence, we can see very clearly. So it means that the PME is generating reactive oxygen species inside the cell, and our probe is able to detect it. So it's at the same time, we can see that the fluorescence is inside the cell. It means that our probe is cell permeable, it goes inside the cell, and then it detects the what is called superoxide. So this particular work we publish in Sensors and Actuators. So not only one probe for the superoxide, we have synthesized different kinds of probe based on the body P. You can see here that we have two body P in one molecule. We have one body P and SCPH at ortho position. we have connected direct SCPH to the BODP molecule instead of using a spacer. Again, we have used two SCPH instead of one SCPH. We have used selenazole molecule, then cyclic diselenide molecule. And what we observe in all this particularly when you have a 2 SCPH, this is giving a very good detection limit, that is 31 nanomolar detection limit. So even very small KO2, that is superoxide, if it is present, then also it can detect. The novelty of this particular work is that if you have one selenium, it quenches the fluorescence of the fluorophore little bit, but if you have two selenium, then it will completely quench the fluorescence of this particular fluorophore, and it gives a very good response after reaction. Not only this, we have synthesized some more molecule, that is disulfide, BODP-based disulfide molecule, cyclic disulfide. We have synthesized coumarin-based molecule, then the quinoline-based molecule, and you can see that in the coumarin-based molecule, this particular molecule is giving a 32 nanomolar detection limit. In quinoline-based molecule, again, ortho will give a good detection limit than the para ACPH. So we have published all these papers in different journals. Now it's time to move ahead with some different analytes, and yes, we have synthesized another molecule, that is BOP. Now, earlier we had only one BF2 units in BODP. Here in BOP, we have bis difluoroboron. That is, we have two BF2 units in the same molecule, and the molecule is symmetrical, you can see. So this is a BOP molecule which is containing with the two BF2 units, and because of that, it gives a very good fluorescence, very high response actually. And we synthesize this particular basic molecule, and then we have gone for the one equivalent of PHACCl, which gives this only one site is substituted, right? And if you use in excess like 3.1 equivalent, and then both the site get substituted with the SCPH. Let me tell you again that if you have one selenium phenyl group, then the fluorescence of this particular BOP molecule quenches, but definitely not that much. And when you have a 2 SCPH, the fluorescence is completely quenched because selenium immediately donates the electron to the fluorophore, and the fluorescence of this particular BOP molecule, which is highly fluorescent, is quenched. Now, once you synthesize this, we have characterized this with the all common NMR techniques, then mass, IR, and finally it's time to get what is called to study the photophysical properties. We were fortunate to get actually the crystal structure of this both compounds, and this you can see here. So we got a crystal structure of these two compounds with single crystal XRD, and you can see that this particular BOP molecule is completely planar, and whatever you have ACPH is exactly perpendicular to this particular BOP plane. Here, in case of the two SCPH, they are exactly trans to each other. That is, one SCPH is above the plane and other SCPH is below the plane. So that is what we have observed in the crystal structure. Now we check for this what is called spectroscopic properties of this photophysical properties of this particular molecule. We observed the absorption maxima at 452 nanometer. And then when we check the emissions with different ROS, here you can clearly see that the emission with the one SCPH is, this particular molecule itself is giving slight emission, right? So there is some fluorescence with this. But when you have a two cell SCPH on the same molecule, the fluorescence is completely quenched. So when you have completely quenched molecule, there are maximum chances to give a very good response, and that is what we have observed. So this molecule particularly gives response to the hypochlorite, you after screening all the reactive oxygen species. Let me tell you one more thing, that we have not only screened the reactive oxygen species, we have screened metal ions, we have screened RSH, that is all biothiols and amino acids. So I'm showing the positive results in the same family, since this is giving result in case of the hypochlorite. So I'm just showing the results of the ROS. But yes, we have screened with all three, that is metal ions, amino acids, biothiols, and the ROS. So here in case of the first molecule, you can see it gives a 3.5-fold increase in fluorescence. However, the second molecule, which is with 2 selenium, gives the 10-fold increase in fluorescence intensity. So after that, we have checked whether this molecule work in physiological conditions or not. So we did the PS study, and yes, we can see it gives a good fluorescence in the range of 6 to 8. Here also we can see it gives a good fluorescence in the range of 6 to 8. So yes, they are working in physiological condition. Now it's time to see the kinetics, whether it gives response faster, whether the whatever response we are getting, whether it is stable for enough time or not, because that is very important when you go for the biological study. So we can see the first molecule gives a response very fast, that is within less than a second. You can see directly start from 0. So response is very fast for the when you have 1 SCPH, and the second molecule gives response in the 10 minutes. And once it gives response, that is once it gives a fluorescence, it is stable for long time. So yes, these molecules are good for the biological study. We can go ahead. This is the detection limit. We did the increasing concentration study, and then draw the graph, and we found that in the first case you have a 1.08 micromolar detection limit. In second case you have a 0.63 micromolar detection limit. So detection limit is also lowered in case of the 2 SCPH. Interference spectra, you can see that we have screened with the other ROS, that when we have probe and KO2, and if you have other ROS present in the same solution, so whether they are doing some interference or what, there is no interference, almost no interference from other reactive oxygen species. The next is cell viability. So again, for this we have choose the MCF7 cell, that is breast cancer cell line, and you can see that first and even both molecules have almost no cytotoxicity, more than 80% cells are viable. So we can go for the next study, that is for the what we call the cell imaging. So for cell imaging, the first molecule here, you can see different panels. So in the first panel, only the probe is there, so there is no fluorescence. The second panel, then the external NaO cell, that is hypochlorite, we have added since it is selective for hypochlorite. Again, no fluorescence. But when you first incubate the cell with the hypochlorite and then add the probe, we can see a very good fluorescence. It means it is detecting the exogenous the NaOCl. And if you add GSH on that, then the fluorescence is decreased. It means that the probe is reversible with GSH. That when you take a probe, add NaOCl, it is giving response, and you add in the same solution, you add GSH, so it will quench. So probe is reversible with the GSH. In the second diagram also, this is kind of endogenous. So endogenous means you added the PMA, which is a natural molecule which will generate the reactive oxygen species inside the cell, not exogenous addition of NaOCl. So, and then it can, if you incubate with the probe, it is showing a fluorescence. Again, with the GSH, it is quenching the fluorescence. So reversibility with the GSH we have observed. We can see in the second case also, same thing. So yes, this particular probe is cell permeable, both the probes, and they are able to detect exogenous as well as endogenous HOCl inside the cells. So this work again we have presented in Analytica Chemica Acta. So for hypochlorite also, not only one sensor, we have synthesized different molecule. Here you can see that the 1-phenylselenide, 2-phenylselenide, and these are symmetrical molecule. However, there is another molecule called BOPI. In this BOPI, one side you are having what is called pyrrole ring, other side you are having a pyridine ring, and 2 BF2 units. So this is unsymmetrical molecule, and this molecule we have synthesized, this BOPI molecule, and this also we observe that this is giving a selective sensing for the hypochlorite, and we can see the detection limits is again lower as compared to the other two BOPI molecule. Okay, it's time to move ahead from the specifically hypochlorite to the hydrogen peroxide, because there may be a different species in the cell, so we should be able to detect specifically. So we need a probe which will detect a specific analytes only, specific species only. So this particular molecule, which is cyclic diselenide, you can see here cyclic diselenide we have synthesized, and when we screen it with the different kinds of ROS, you can see that this is looks like a 5 or 4 legs or 5 legs. Participants, elephant with 4 legs or 5 legs?
5.
Sir, 5 legs.
5 legs?
5. Yes, sir.
I can see here with the
Only 4 legs, sir.
4 legs. So it's kind of illusion kind of things. So let me tell you that many times chemistry also confused like this. That sometimes we design something and we think that we'll get this, we get something, some different results. So it becomes difficult to analyze the results, and that is what happens in this particular case. This molecule that we synthesize the cyclic diselenide, we thought both selenium will get oxidized and we'll get a response with the both selenium. And what we observed that, okay, this is a just characterization spectra. We have characterized this. So what we observed, this particular molecule is giving response to the H2O2, not to the superoxide, not to the hypochlorite, as we assume that it will give to the hypochlorite or superoxide. But yes, this is giving response to the H2O2. And we can see response is almost 9-fold increase in the fluorescence. In the diagram, we can see the fluorescence also. The molecule without H2O2, the molecule, the probe with H2O2 is giving a very good green fluorescence. We check we did the PS study. It is giving response to in the physiological range. Then the what is called interference study. This is also give a good result. Then we did the detection limit, and what we observed that this molecule also gives a very low detection limit, that is 44 nanomolar detection limit. And yes, it is giving a fluorescence at start very high, but slightly it is decreasing, but still it is enough fluorescence to detect inside the cells. So this particular molecule we synthesized, we did the study, and we presented this particular study in chemistry and ASEAN journal. Okay, we did a lot of study on the reactive oxygen species. Now it's time to move ahead and design something which will react with the metal ions, which will trap the metal ions. And that is why we have designed a core where you have 2 tellurium and 1 nitrogen. So this is a kind of, you can say, a pocket where the metal can go and sit. So this can coordinate with the metal, and we can detect different kinds of metal ions. For this, we have synthesized this particular BOPI molecule. We have done almost 1 year work on this because the strategy were not working out. That we tried to synthesize first BOPI and then tried to have a bromine on that to attach this particular pocket, tellurium natrium pocket to that BOPI, but that was not working. So we first did the bromination, and then after bromination, we have attached this particular molecule and we were able to get this molecule that is with the 2 tellurium and nitrogen packet. Okay. So we synthesize this molecule, we characterize with various techniques like NMR, IR and all those common techniques. At the same time, we were lucky enough to get the crystal structure, and you can see the crystal structure of this particular molecule. We have 2 tellurium and 1 nitrogen. And 2 tellurium, they are far apart from each other. They are on the different sides when the molecule crystallizes in solid. So, but let me tell you that whenever it will be in solution, there will be a flexibility, and when the metal ion comes, it can coordinate with the metal ions, and that is what happened. So after characterization, we did since this molecule was designed specifically for the metal ions, but we did the study with all, that is ROS, metal ions, and amino acids. And this particular molecule, you can see in absorbance spectra that this is giving very different response with the mercury, that is Hg2+. With all other metal ions I have quoted here, there was almost same kind of spectra. And when we check in the emission spectroscopy, what we have seen that this particularly with the mercury, it is giving a very good response, a distinct response than the other metal ion species. So 2.5-fold increase in the fluorescence, and it was giving almost more than 50 that is shift from the absorbance to the emission, right? So absorbance was at 486 and we got emission at 547 nanometer. So after this, we did pH study. Yes, it is giving a response in the physiological conditions. Then all other metal ions should not disturb this fluorescence, because if they disturb the fluorescence, then we will not be able to detect a particular metal ion selectively. And that is why interference study becomes very important, that interference of the other metal ions should not be there. Okay. So we did the interference study and we can see that almost there is no interference of other metal ions. Yes, it's time to do the kind of time dependent study, what we call the kinetic study. And this gives again a response within a second. And detection limit has been observed to be 62 nanomolar. So this particular compound gives a very good detection limit. That is, if mercury is present even in lower concentration, that also can be detected by using this particular probe. So once you know that it is detecting, but in what is the ratio of detection? Whether it is 1:1, 1:2, or what? So what we did we did the study with the different concentration and plotted a job plot. So you can see here from the job plot, it is very clear that the probe and metal binding ratio is 1:1. That is, one metal is binding to the one probe molecule. And that is what we were expecting, that one metal ion will reach into the pocket of this particular tellurium and nitrogen and it should give a response. So yes, it is working. And when we did the reversible study, so what we observed that when you have a probe and mercury, it gives a response. And in the same solution, when you add different kinds of biothias, the response is quenched. So probe is reversible with specifically with the Na2S, not with all other biothias. You can see homocysteine, cysteine, GSH, N-acetylcysteines. It is the fluorescence is almost not disturbed, but with the Na2S, it is quenching. So the probe is reversible with Na2H. And what we can do in the same solution, you again add mercury. It will give again fluorescence, and in the same solution, when you add again the Na2S, the fluorescence is quenched. So this cycle, reversibility cycle, it moves up to the 3 redox cycle. That is what we observed. This is the sensing mechanism, that how it has been detected, how particularly we are getting response for the mercury. So what we observed that this particular mercury is going inside this particular pocket and it is giving response. This we have proved with the NMR, that these two CH2 peaks, they are shifted in the NMR after reaction. And at the same time, we did the mass spectra, and mass spectra, we got a M plus peak. So with the isotopic pattern of the mercury and tellurium, because isotopic pattern in the NMR is very important, and it gives you the idea whether your particular element is present in that or not, if it has isotopes. This is the PET mechanism. Again, you can see this is in quench state, and and when you add mercury, it is giving you the fluorescence. Now it's time to go for a biological study. This particular molecule, we observed that we did the cell viability study. Slightly toxic it is, but yes, it can go inside the cell and it can detect the mercury inside the cell. That is what we have observed. The probe itself, because since probe itself was having some fluorescence, so some fluorescence we can observe here. But with mercury, there is no fluorescence. And when you have probe and mercury, it gives very good fluorescence. And fluorescence itself is located inside the cell. It means this particular probe have cell permeability also. So this work we presented in Dalton transaction. So not only one probe, we synthesized different probes. You can see this diselenide probe. Actually, this was a completely unexpected result. That is why many times chemistry work unexpectedly. So this we synthesize the diselenide probe. We first thought that it will give a response to the biothiols. It will break down the ACSC bond. But this particular probe gives very good response to mercury. It breaks down the ACSC bond and rearrangement takes place, and this particular molecule was formed. First, we were not sure about this, since even though we got all spectroscopic data, but finally we got a single crystal structure for this molecule, and yes, the rearrangement takes place. So again, we have synthesized rhodamine. Again, you can see a diselenide based rhodamine. We have a single crystal structure for this diselenide based rhodamine molecule. And it is again detecting Hg2 plus with very good red fluorescence. You can see here. So almost near our fluorescence there for the molecule. There is some hindrance from some. What is called Vijayanan English? Please, can you mute here? Yeah, thank you. Okay, so this is a mechanism again for the detection of mercury by the rhodamine based probe, and we have proved this and then presented, published this paper in another Dalton transaction. So in comparison, we have synthesized 3 different tellurium and selenium based probes, and we can see that this the first tellurium based probes with the pocket gives a very good detection limit, that is 62 nanomolar. Okay, it's time to go for another one. Now we move to the another one, that is sometimes when you see at the same molecule in a different way, with the slight change, so you can get a different results like this. Somebody will say that yes, this person is looking just straight on the what we can say right direction or in the front direction. This kind of illusion. So in the same way, we synthesize this particular pocket with the selenium, since we had experience with the tellurium, and we thought it will again detect the mercury, because selenophilicity of the towards the mercury is well known, and so we thought it will also give response to the mercury, but let's see. Even though I have mentioned in the title that it is for copper 2 plus. So we synthesize this with the whatever earlier strategy we had. So we got this particular probe in 33%. You can see that instead of tellurium, we are having 2 selenium in the pockets and a nitrogen, and characterize this with common spectroscopic techniques, and then we have gone for the photophysical study. So here you can see in the UV spectra itself, this particularly is giving response to the copper 2, that there is a change in the absorbance. And when we check in the emission with all other metal ions, there is no response, almost no response. With one metal ion, that is, I think, Ag plus, with small response was there. But with other metal ions, there is no response. But when you add copper 2 plus, it gives a very good response with the 6-fold increase in fluorescence. We did then what is called the pH study. Yes, it is working in the physiological pH. There is no interference from the other metal ions. That also we observed. Instant detection of the copper. You can see a very fast response to the copper, within a second, you can say. And detection limit also, you can see it comes down to the 10 nanomolar. So detection limit almost is 10 nanomolar in this particular case. And again, Jobs plot, as we discussed in case of the mercury, it gives 1:1 ratio binding of the copper and the probe. It is reversible again with the Na2S, not with the all other metal ions, but just one cycle, not more cycle, because it maybe after that the probe is decomposing. I think means we assume that we try to find out the traces, but it was not observed. So this particular molecule can sense the copper. Again, you can see the sensing mechanism, that if this particular copper goes into the pocket, and why in case of the tellurium, mercury is going in the pocket, and in case of the selenium, copper 2 plus is going in the pocket. That this depends on the size of pockets and the size of metal ions. That is what we believe. We have observed the change in these two CH2s, that there is change that is shift in the peak. And same thing we have observed in the mass also, that yes, we got the M plus ion peak in the mass with very good isotopic pattern of the metal ions. So this particular sensing mechanism, again, we proved is due to the PET, that is photo-induced electron transfer, and it gives a turn on fluorescence. Since title of my particular presentation was to detect the cancer, and that is what we try to do in all cases. So in this case also, we check first the cytotoxicity and then bioimaging. You can see in bioimaging that the particularly copper, when only copper is added, there is no fluorescence, or when only probe is there, very small fluorescence. But when you incubate the cell first with the copper and then with the probe, you can see a very clear fluorescence. And here you can see when you have a merge diagram, then we can see that the fluorescence is inside the cell. So again, our probe is cell permeable. It can go inside the cell and can detect the copper. So this is what the output of this particular work was. We have published this in Journal of Molecular Structure. So let me conclude the research work. We are working on different kind of selenium tellurium based molecule for the detection of the environmentally and biologically important analytes. Specifically, what I have mentioned, the malate cascades, metal ions, ROS, and RNS. And we did a lot of work. I think more than 30 molecules we have synthesized. We publish in different papers. So all these are superoxide sensors. These are superoxide sensor. Here you can see hypochlorite sensors are also present. And then the mercury ion sensors and copper ion sensors. And there are many more. I have selected papers I have shown here. We have one accounts of chemical research also on the what is called selenium or tellurium containing sensors. So that is published in 2014. Okay. There are a number of papers in the what is called in the pipeline. Some accepted, some are in revision stage, and so on. So let me thank to the contributors of this particular work. So you can see this is my lab. When my postdoc Professor David Churchill, visited our laboratory in the Department of Chemistry, University of Mumbai. Particularly, the work presented here was done by Pratiksha Deshmukh, Gauri Malankar, all these are Dr. Divesh Shalar, and some work is done by Dr. Sri Krishna Salunke. Then Mr. Kasinath Nadibuni, they have submitted he has submitted the thesis, however is not it done. And then Mr. Miss Snell Salunke, she is in the verge of submitting the thesis. There are many other students. Let me thanks to the collaborators, that is S.K. Maji for biological work from the IIT Bombay, R.J. Butcher from Howard University, USA for giving us crystal structure data. Then Professor Rahul Pinjari, right now he's working in ICT, earlier in the Nanded University. So for giving us computational chemistry support. And Professor Malay Patra from TFIR, again for the biological study. For funding, I would like to acknowledge DST SRV for the Young Scientist Award, UGC, and other agencies for the financial support. Definitely Department of Chemistry, University of Mumbai, friends and family. There is 1 year extra in my family. So by mistake, it was happened during my PhD presentation, but I have kept it always because they had pointed out that there is 1 more extra year. So I said that I have a very good or kind of strong support from my family, and that's why 1 year is extra. So I keep it always there, even though it is spelling mistake. So this is my current laboratory, and this is current laboratory. You can see these are the current students who are working, but just 1 month before we have shifted in this new lab. Right now we have a new lab now. From students are working in this lab from last 1 month. This was just when we are going to shift in the new laboratory. Okay, now it's time to change the subject. I think we have half an hour more, right?
Yes.
Yes.
Thank you.
Yes, sir.
Thank you.
Yes, sir.
You can, till that you can observe the diagram and see the illusion. I had a few sip of water. So thank you. Okay, lot of chemistry, actually research chemistry. Let's go to, let's change the topic now. It's really sometimes the mind gets saturated with the same kind of things. So let's change the topic. We'll go to the introduction to the isolable analogy. Now this is the topic I teach to the master students, second year.
Can we have question before that, if you
yes, please. Yes, please. Sure. You can have.
Yeah, just I have one question. So regarding the cytotoxicity of the species, the selenium dioxide, which you have mentioned in the first itself, the first chapter, you have showed the cell cytotoxicity study.
Sorry, your voice is breaking. Can you repeat it again?
Can you hear me?
Now.
So, saying with this selenium connected to the body pie. So there the selenium dioxide is there. So generally, what I have seen till today, maybe I am wrong. So selenium is useful in the neutral state mostly.
Yeah.
It is in +4 oxidation state, and you have shown only one study to claim that this is safe. So is it?
Actually,
we need to do some biocompatibility study or some in vitro or pharmacokinetics.
Yeah, that is true. But right now, this technique has gone advanced. So selenium earlier it thought to be toxic, then it becomes an essential nutrition. And nowadays, when we have studied particularly these molecules, the cytotoxicity, the molecule was not giving cytotoxicity at all, in particularly selenium case. In tellurium case, yes, there is some cytotoxicity, but in selenium case, we have not observed, and we are using in very small concentration. The concentration of
My
this
question
in,
is that only study is enough, or we need to do some more study?
we need to go. We need to go for advanced study. We need to go for
because
advanced.
by reading claiming the cytotoxicity 90% safe with a single study, I think it is.
No, definitely we need to go for advanced study. Yes.
Yeah,
I agree. Yes. Agree. We need to go for advanced study. Yeah.
Sir, hello.
Yes, please. Yeah.
Sir, superoxide. How to stable the superoxide ion?
Sir, here we use commercially available KO2. Okay. And we use this in DMSO. But here you have to use freshly made superoxide. You dissolve it and immediately use it. You cannot keep it for longer time. Otherwise, again, it will decompose. And decomposition rate is very fast.
Okay, sir. I think, sir, are you use NPTC, sir?
Huh?
Are you use NPTC? Are you use NPTC?
No.
Which I say?
We use especially MTT.
Hello.
MTT. Okay.
Sir, your voice is breaking. We use MTT assay for this.
Sir,
okay.
In one of your presentation, you say that the biological evaluation of fluorescent probe for detecting the mercury second, that is mercury ion, in case of this human cervical cancer cells. So my question is that if this probe is highly selective and biocompatible, what are the major challenges in translating it from that human cervical cancer cells, imagining to clinical diagnosis of
the major
mercury pyogenesis?
As one participant asked the question that the advanced study is needed just for cytotoxicity, because the probe itself should not be more cytotoxic, even though we did it in cell study, but we have not done in the animals. So we have to go for animal study, and we have to check whether there is cytotoxicity of this particular molecule in the real animal study. That is most important. Even though, yes, we are very much confirm about that this will detect the particularly metal ions inside the cell, but the cytotoxicity issue remains.
Yes. Okay.
Hello.
Yes, ma'am.
Sir, are these sensors soluble in water, 100% soluble in water?
Some are. Actually, we used to dissolve them in small amount of DMSO and then dilute it with the water. Some, I will say most of them. Only few probes they are not soluble directly in the water. So we have to use 50% DMSO and 50% water. But in most of the cases, we use just small amount just for the solubility of the particular 2 milligram or 3 milligram of the probe in 1 or 2 drops of DMSO, and then you dilute it with the buffer solutions or water.
So the use of DMSO will not be a problem at physiological conditions?
Actually, just for dissolution, that much concentration is okay. That is accepted.
Okay.
Hello, sir. Hello.
Yes, sir.
Sir, I have also said one question.
Hello, sir. Two people are talking. Can you tell me who is talking first?
Sir, hello, Pawan Kumar, sir.
Anil Kumar. Anil Kumar, please ask.
Sir, during the addition of superoxide, any color develop in the
No. If there is a visual detection that if there is a color change directly in some particular probes, there is a change in the color, but in some cases there is no change in color. There is no change in color. Yes, another one?
Sir, I am Pavan Kumar.
Yes, Pavan.
Sir, I'm asking that sir, which in the the metal ion you used in your study, sir, in the which form, sir? It is either, sir, carbonate or
No,
chloride or it's
perchlorate. Yeah,
perchlorate. Sir, TBA, tertiary butyl ammonium, or say in the form of perchlorate, sir.
Yes, I think most of them are in the perchlorate. Just maybe
Okay, sir.
one or two in different, yeah. But most of them are in the perchlorate, and we use Aldrich, all the Aldrich standard means metal ions.
Sir, one ma'am is asked from the you study have done in solution with solution with water. So the water or maybe sir in buffer solution also.
Yeah, buffer solution. In some cases, buffer solution. In some cases, pure water. That is double distilled water we use.
Okay, sir.
Yeah.
Thank you, sir. This is my question, sir.
But whenever we did in double distilled water, for pH study, we specifically use the buffer solution.
Yes, sir. Buffer solution, sir. Good, sir. Thank you, sir.
Any other question, please? If no, then can we move ahead?
Yes, sir. Sure, sir.
Yes, sir.
Thank you.
Yes, sir. Yes, thank you.
Okay. So as I was discussing that this is particularly I'm teaching to the master student, that is isolable analogy. So let's first we'll go that why this isolable analogy is important. So as we know that there are different methods to find the structure of the clusters. Okay. So by using the effective atomic number, or we call it, in case of the clusters, we call it magic number. In case of the monomers, we call it as a what is called 18 electron rule. Okay. So they are important. And based on the this valence electron, that is the effective atomic AN or magic number. So based on this number of electrons, total contribution to the monomer, dimer, trimer, we can predict the structure. So if you have total valence electron, then 18, then monomer, and 48 triangle, you can have 60, then tetrahedron, and so on. So it is easy to find out this electrons. We can see the examples that if you have iridium 4CO12, then we know that iridium is a this is called D7S2 system, and that is total 9 electrons. So there are 4 iridium, so 9 into 4. CO, that is carbonyl group, will give 2 electrons, so 2 into 12 and total 60. So we can say 60 means tetrahedron structure. Similarly, AP5CO15 and C. So this AP will give total 8 electrons. So and CO, each CO will 2 electron, and carbon, which will give 4 electrons. So total 74 means square pyramidal structure. And in this rhodium 6 and CO16 case, you get 86 total and octahedral structure. So this is one method to predict the structure of the clusters. This is the prediction of the structure of the clusters. And we can see this examples, that is AP3CO12, which is 48 total valence electron, and the structure is like a trigonal. Okay. Even with if you some of the COs, they are bridges, and some of them are what is called terminal. When you have larger metal ion like ruthenium and osmium, so all the CO goes as a terminal, no bridging is there. And even after that also we have what is called the trigonal structure. In case of this particularly CM, that is tetra nuclear metal, that is M4CO12, you can see this gives a 60 number and again tetrahedron kind of structure. When you have larger atom, you will have all COs as a terminal. And in case of this, as we discussed, that is rhodium or even cobalt, they will give you a octahedral kind of structure with the formula M6CO16. So this is one method to find out the structure of the molecule. Now, when we come to the isolobal analogy, what does mean by the isolobal analogy? That is molecular fragments, 2 or more. We call them isolobal if the number, symmetry, energy, and shape of the frontier orbitals in them and the electrons occupied by them are similar. So what does it mean? That whatever the particular frontier orbital is there, that should have same energy, same number of electrons occupied them. Symmetry also need to be same. Okay, this was observed by the Roald Hoffmann in before 1981, and he got a Nobel Prize in 1981 for this particular work and another work. Now we will try to understand what does it mean. That here we can see that CH3 looks like isolable. This is the symbol for isolable analogy, is isolable with Ml5 system. So CH3 we always call a radical, means with the 1 electron. So there is a 1 orbital with 1 electron, and similarly a Ml5 system will also have. But Ml5, which Ml5 system? D7 Ml5 system. Because if you take a D6 Ml5 system, then there will be no electron in that frontier molecular orbital. So that is like a plus CH3. Okay, we'll go in detail. Time is there. So we'll go in detail. So let's see that how this frontier molecular orbital generates first. So this is an octahedral diagram of this Ml6 system, that is 1 metal and 6 ligand. So we can see that all 6 ligands are donating electrons, and it forms a 6 molecular orbital. And you have T2G electron as it is, which lies here. Okay. And if you remove 1 ligand from this, then what happens? That if you remove 1 ligand, you have a D6 Ml5 system. The first was D6 Ml6 system. Now you have D6 Ml5 system. Structure will look like this. So you have only 5 ligand which are donating electrons into the 5 hybridized orbital, and it forms 5 molecular orbitals with the pair of electrons. T2G orbitals, they are as it is with the T2G electrons, 6 electrons. The 6th orbital, which is not occupied by the ligand, it will come here, and which is empty. Here you can see. And that is called the frontier molecular orbital. So this particular molecule is having 1 frontier molecular orbital with no electron. Now, let me tell you that if there is no electron in that orbital, then this cannot make a bond. This cannot, even though it is there, it cannot make a bond with kind of sharing. So you should have at least 1 electron into that, then only it can share. Otherwise, ligand will come and donate. So it will go back again like D6 Ml6 system. Okay. So with this, you can see that if you have a molecule like CrCO6 and you remove 1 ligand, it will become CrCO5 with 1 empty orbital, frontier molecular orbital with no electron. So similarly, if you compare it with the organic fragment, that is CH4. So CH4, if you remove 1 hydrogen with 2 electrons, then it will become CH3 plus, and you will remain with the CH3 plus, means 1 frontier orbital with no electron. And we know that CH3 plus, they cannot form, or with the similar fragment with the same molecular orbital, it cannot form the bond, because the other one will also have frontier molecular orbital with no electrons. So there will be no bond. But if you have a system like this, that is Ml5, D7 Ml5. D7 Ml5 means you have 6 electrons which are going into T2G and 1 electron which will go into this frontier molecular orbital. So you will have this type of molecule where there is one frontier orbital with one electron, and same case if you check in the organic molecule, you will have a CH3 again with radical that is one electron. So we can have CH3 and Ml5. Now these two are isolable. Ml5, that is the example, best example is MnCO5. MnCO5 and CH3, these two fragments are isolable because MnCO5 we are having 5 ligands, and in Mn we are having 7 electrons. Out of that, 6 goes into T2G and 1 into the particularly this frontier molecular orbital, and it is available to form the bond. So this MnCO5 can form a bond with CH3, and yes, we know that it exists. MnCO6 is not there. MnCO5 itself is not stable, but Mn2CO10 is stable, and the reason lies into this only, that Mn2CO10, that is there is a Mn-Mn bond, because one MnCO5 and other MnCO5 they are having two frontier molecular orbital, and they can get connect and form a bond. So similarly, CH3 and CH3, one CH3 can connect to other CH3, and we know that C2H6 is well-known ethane molecule. So if you remove one ligand, you will have ML5 system with one frontier molecular orbital. If you remove two ligands, and system is FeCO4. So if you remove two ligands, then you will have a two frontier molecular orbital, and system is D8. D8 means again 6 goes into T2G and 2 goes into this two frontier molecular orbital. The same similar organic fragment is CH2, where you can see that CH2 with the 2 electrons can have 2 frontier molecular orbital occupied with one electron. And yes, we can connect 2 CH2, we can get C2H4 molecule. We can connect even 3 CH2, we can get a cyclic cyclopropane molecule. Even though with the FeCO4, double bond FeCO4 doesn't exist, but yes, they can connect with the CH2s. Those kind of molecules, they exist. So here we can see that molecular fragments can combine into molecules even between the groups, and all of them compounds shown below are known and stable. So we can see CH3, C2H6, even the Mn2CO10 is well known. This MnCO5 and CH3 can combine, and it can give a CH3MnCO5. So if you have two frontier molecular orbital, then this type of molecules can be formed. So with cyclic molecules. Now, if you extend this analogy, so we can say that this particular MnCO5 is isolable with CH3. Now, Mn, the difference between the Mn and Fe is 1 electron. So we can write FeCO5, but with plus charge. So they are isolable. And finally, if you see that MnCO5 and CrCO5, there is one less electron into the Cr than Mn, so we can write CrCO5 minus. Okay. So this kind of isolable analogy we can develop. We can develop more, that is, you can see that CH3 minus, which is isolable with FeCO5 0, can be isolable with CrCO5 2 minus. Okay. Even CH3, which is we call the radical, that is isolable, as we know that this radical is isolable with MnCO5. Here we have shown that MnCO5 is isolable with FeCO5 plus and CrCO5 minus. So in this way, we can develop the isolable analogy. Extending this to the other molecules like MnCO5, that instead of CO5, we can take similar donating, that CO donates 2 electrons, so PR3 also donates 2 electrons. So we can change this. You can take Cl minus also can donate 2 electrons. as we know, Cl will have only one, but Cl minus will have 2 electrons. So it can, even Cp and benzene ligands also can be incorporated with the 6 electrons, that is 3 pair of electrons. So that also we can include into this and can be converted into the different kinds of fragments. Okay. I'm not going into so much detail because of times. It's already just I think 10 minutes or remain, so I will just try to wind up faster. So we can have different examples of isolable analogy, that this particular CH4 is isolable with CrCO6, or CS3 is isolable with MnCO5, or this Fe and Fe. If we kind of convert it into the CP cyclopentadiene ring with the 3 COs, and in the same way. Okay. So these are different molecules which are isolable with each other. And how to use this particular isolable analogy to deduce the structure, that we'll see. We'll take one example of this one, that we know that there is another method, what we call the Wade's rule, to find out the structure. That if you Wade's rule says that negative charge contributes 1 electron, hydrogen contributes 1 electron, BH units contribute 2 electron, and CH unit contribute 3 electron to the skeleton. And based on the number of electron pair, we can predict the Wade's type. Like n-1 number of electron pairs, then super hypercloso, or n+1 if it is there, then closo, n+2 nido, and so on. Okay. So with we'll see one example, few example. I know that all of you must have known all these things, but still I want to go through this because from that only we have next example based on all this. So if we have a B6S62 minus cluster, then the n will be the 6, that is number of boron present in the particular clusters. And we, as we learn, that each BH unit gives 2 electrons, so total 6 BH units, 12 electrons. There are 2 negative charge, means 2 electron. Total electrons will be 14. So 7 electron pairs, and we can write in the form of n, that is n equals to 6, means 7 we can write 6 plus 1, and ultimately n+1 closure structure. So others are also same with the you can see here n+1 closure structure, or if you get n+2 nido structure. Here in this particular example, B is that is boron is n equals to 5. In this particular case, we count the number of boron and number of carbon. So we have 2 carbon and 10 boron, so n becomes 12. And we know that CH gives a 3 electron, however BH gives the 2 electrons, so you can count it. Now based on this, when we apply all this to the cluster, that is organometallic cluster, so we'll take one example, that osmium 3CO12, if you hit it, it gives a different kind of clusters. And if you see the osmium 5CO16, if you try to find out the structure of this particular cluster, so we can try to understand it from the isolable analogy, that we know that CO3 can be isolable with CH, as we have seen from here. Here we can see that CH is isolable with CO3. Okay, and so this CO3 is isolable with CH. We can write in the form of BH minus, that is 1 electron. Boron has 1 electron less than carbon, so we have given the minus charge. So BH minus, and as we know, CO means cobalt. Below that we are having osmium. So we are having osmium CO3, which we can sorry, not osmium. Osmium is just below the iron, which is 1 electron less than the cobalt. So instead of BH, we can BH minus, we can write the BH. So osmium CO3, which is isolable with BH. Now we have, once we have converted this into this particular isolable analogy form, we can use this in the osmium 5CO16 molecule, which we can write, that is 1 CO means 2 electrons, So I have written osmium 5 CO15 and H2 because I want to convert this osmium CO3 into this. So osmium CO3 by 5, it will become osmium 5 CO15 and then 2 electron with the H2. Ultimately, it will become BH5 and H2. And then we know that B5S7 means total, it will give 6 electron pair, and we can, in this way, we can deduce the structure of the osmium 5 CO16, that is as a closure. So in this way, we can apply this analogy to the different molecules. There are, again, definitely you need to keep in mind a lot of things like number of electrons, in which group it lies. Like this example was, I think, in the NET exam, which year I don't remember exactly, but this was in this I have taken from the NET exam. That there was a question that if you have a Bi5 3+ cluster, and if you add 2 electrons into there, then what kind of weights type cluster you will get, that is from which to which. So there were so many options. So we can see that bismuth is present in the nitrogen family. So bismuth and nitrogen, and we know that CH means 3 electron. So NH 1 electron more than CH, so 4 electron, and nitrogen definitely will have a 3 electron. So similarly, B5 will have 5 into 3, total 15 electron, but there is 3+ charge means 3 electron less, so 12 electron, so 6 electron pair, and 6 we can write in the form of N is 5, so we can write N+1, and that is closure. If you add 2 electrons into this, definitely it will go to +1, and you will have 7 electron pairs N+2, that is nido. So this goes from closure to nido. So this such type of questions appears in NET examination. Organometallic chemistry, I teach a complete paper in the 4th semester of the inorganic students, and this is one of the best books to teach the organometallic chemistry. What I found, definitely there are a number of other books, but I found this is one of the best book. So thank you very much. With this, I think I have almost, it's 1:42. With the research on my academic, I like to do farming, so I do a lot of farming. So I will just go through a few photographs. You may like this. I do a lot of farming at my hometown. Mango, custard apple, then guava, and different types of plants are also available in my farm. So thank you very much for your kind attention. Yes, any question, please?
Thank you, sir.
I know that I have taken introduction to isolobal analogy very short, but just I wanted to touch the topic,
Sir,
that if somebody is unknown with the topic, you people will come to know. Yeah.
Sir, I have a question.
Yes, please.
Does this isolabel analogy applies only for carbonyl ligands?
Isolable analogy is applied only for carbonyl. No.
Does this
similar kind of you can use the similar kind of ligand. You can use this isolable analogy. You can use for CP. Even we have used PR3. Just you need to convert it. That number of electron, how many number of electron pairs it will be donated by that particular electron. So basic, we are taking a carbonyl compounds. For basics understanding, we are taking this carbonyl compound. Then you can definitely move on. Because it's easy to understand from the carbonyl compound, and for actually the students where we are introducing this to the MSc student, so it becomes for them to understand this new topic easily with the easy examples. There is a lot, but because of short time, I have kept very specific example. There is a lot to study in this. So we used to take almost 15 lectures to complete this isolable analogy. And each examples you need to explain way in lot of detail. I cannot do that in this short time. Yes, any other question, please? If not, then we can stop here. Thank you very much for your kind attention. Yeah. I think I am done with the time.
Good afternoon.
1:44. We are on time.
Yes, sir.
Thank you.
Sir, I am audible, sir.
Yes, please.
Good afternoon to everyone. Myself, Birbarao Gujabrao Maske, Assistant Professor BBArt, NB Commerce, BP Science College, Digras. On behalf of all the participants, I sincerely express our heartfelt thanks to today second session resource person, Dr. Sudesh Manjre sir, for delivering such an informative and engaging session on new topic to detect the cancer and isolobal analogy. Sir, your valuable insight, clear explanation, and practical approach have helped us understand the topic better and inspire us to enhance our knowledge in chemistry. I would also like to extend our sincere gratitude to the organizer, Dr. Kodape ma'am, and supporting team for planning and conducting this refresher course so efficiently. Finally, I thanks all the participants for their active involvement which made this session interactive. Thank you again, sir. Have a nice day.
Sir, yeah, please unmute yourself, sir.
Hello.
Yeah. So
Yes, sir.
thank you, Kodabe madam, for inviting me once again. And thank you all the participants for your active interactions and asking the questions. I hope I was able to deliver at least something to you.
Thank you, sir. Thank you. Very informative lecture, sir.
It was really very informative,
Thank you, sir.
Thank you, ma'am. Thank you, all of you. Can I leave now?
Thank you, sir.
Thank you.
Yeah, our next session is going to start on 2:30. Who is going to introduce the next resource person?
Myself, Birmal Mary Matthew, ma'am.
Okay, so be ready at 2:30. So if I will not there, please take care of the resource person, Dr. Yuvika Thakre. So you introduce her and let the session begin at that time.
Okay, ma'am.
Okay. See you at 2:30.
Sure, ma'am.
Hello, am I audible?
Yes,
Yes.
sir.
Yogita Thakre, madam. योगिता ठाकरे मॅडम.
सर सांगते मॅडमला 2 मिनिट.
बरं.
हॅलो.
हा मॅडम सुरू करायचं काय?
हो करा सुरू.
बरं. Participant who have the responsibility for giving introduction.
Yes, sir. Yes, sir.
So madam, please proceed, madam.
Good afternoon. Thank you, sir. Good afternoon, everyone. It's my pleasure to introduce the resource person of this session, Dr. Yogita Thakare, Associate Professor, Department of Chemistry, Sri Shivaji Science College, Amravati. Ma'am took her BSc, MSc, and PhD from St. George Baba, Amravati University, and topic of her research work is extractive spectrophotometric determination of some metal ions using acetyl derivative of calyx ray. She is a recipient of several gold medals, cash prizes, and scholarships when she was a graduate and postgraduate student, and this reflects her academic brilliance. As recognized research guide and with about 19 years of teaching experience, Ma'am took various lectures and also served as subject experts in refresher courses, science exhibition, conferences, and seminars. Special mention to the preparation of Fundamental Glossary of Chemistry in Marathi, a program of Ministry of Education, Government of India. Ma'am's area of research includes supramolecular synthesis, physicochemical, and kinetic study. Ma'am has about 20 publications in reputed journals and presented about 50 papers in various national and international conferences. Author of 5 book chapters in the area of chemical kinetics. She is appointed as VC nominee and subject expert in various selection committees of this university. Yogita ma'am, on behalf of the organizing committee and all participants, may I extend a warm welcome to the session, Electroanalytical Techniques, Polarography, Basic Introduction. Welcome, ma'am.
Thank you for the nice introduction.
You are audible, ma'am. Continue.
Yes. Is it visible, PowerPoint presentation?
Yes, it's visible. Yes, ma'am, it's visible.
Okay. Dear participant, we will start with the. Is there any echo?
Yes, echo is there, madam.
Yes, ma'am.
But why? I don't know. I don't know. Anyway. So welcome you all. This is the electroanalytical technique, polarography, and we will start with the basic introduction. This is a very basic topic in the field of electrochemistry. As a physical chemistry person, I very much like the electrochemistry, chemical kinetics, solid state, and so on. Now, what is electrochemistry? It is simply the branch of science which deals with the interconversion of electrical energy into chemical energy, or either of. Now, in electrolytical cell, the conversion of electrical energy into chemical energy there, and in electrochemical cell, there is a conversion of chemical energy into the electrical energy. Now there is a very confusion. What is meant by the anode and what is meant by the cathode? Now what is anode? Anode is not a positively or negatively charged electrode. Anode may be positive or negative, or cathode may be positive or negative. It depends on which kind of cell is it. It is electrolytic cell or it is electrochemical cell. Now, then what is anode? It may be positive or it may be negative. Now, anode is the electrode on which always oxidation occurs. And cathode is the electrode on which always reduction occurs. Okay, so anode and cathode may be positive or negative depending on the type of cell, electrolytic cell and electrochemical cell. Now, in electrochemical series, all the electrodes are arranged with the decreasing order of their reduction potential. Now you can see here the different kind of metal ions and its reduction potential, and it is in the decreasing order. Suppose here the reduction potential is 2.87, it is plus, and this is the reduction potential, it is minus 3.05. Means it is having the low reduction potential and this is having the higher reduction potential. So if reduction potential is more, it will get reduced, and if reduction potential is less, it will get oxidized. This is simple the electrochemical cell in which chemical energy is converted into the electrical energy. It means because of the chemical reaction, we will get the electrical energy. Now, this is simple zinc and copper electrode. Zinc having the electrode potential minus 0.76 and copper having the reduction potential plus 3.4. Now, copper has the more reduction potential, so it will get reduced. So this is the reduction half cell. And zinc having the lower reduction potential, so it will get oxidized. Okay. If oxidation is there, it means it acts as an anode. So anode is the zinc electrode in this particular cell, and cathode is the positively charged electrode. Okay, copper here. Simply these two half cells attached by this salt bridge for the moment of ion to complete the circuit.
Madam, sorry to interrupt you.
Yes.
Can you make it full screen?
Full screen. Okay.
I am just from the beginning. The current slide.
Yes, is it visible?
Yes.
हाँ, वही तो पहले मैंने पूछा कि दिख रही है क्या? Okay, and echo is there. I don't know why. Okay, so zinc will get oxidized. It is anode. It is negative here. Copper is reduced. Okay, it is cathode, positively charged electrode here. Now, this is the circuit for the flow of electron, and to complete the circuit, okay, salt bridge is added for the flow of ion. So what we have to understand here, that is simply if I want to find out the EMF of cell, it is given by the Nernst equation, and it is equals to E0 minus 0.0591 divided by N log of concentration of product divided by concentration of reactant. This equation, in this balance equation. So E is equals to whatever the product is there, it is Z and I positive if minus sign is here. And copper is the reactant, okay, in this particular balance reaction. Now, if I attach the oxidation half cell and reduction half cell, we will get the total EMF of cell. So there are the two quantities, EMF of cell, EMF of right hand cell, and EMF of the left hand side cell. So if I know the two quantities here, if I attach the right hand and left hand, definitely I will get the EMF of cell. From this right hand and left hand, if I must know the value of one, either oxidation half cell or reduction half cell, then I can find out the EMF of the other cell. So in this particular electrochemical reaction or electrochemical cell, we use the two electrodes in which one is oxidized and one will get reduced. Now what happened here? Zinc is the anode, means oxidation is there. So zinc means this electrode is zinc. Zinc will dissolve because it forms the Z and I positive ions and two electron, okay, oxidation. On the other hand, this is the CuSO4 solution. Cu dye positive from the solution, okay, reduced to form the Cu. So these two electrons from the zinc. So Cu dye positive ions plus two electron, it will get Cu. So this is the reduction reaction. Now if we observe here, this electrode, zinc get dissolved. So electrode will dissolve here, and here copper ion from the solution will get deposited from the copper ion from the electrolyte solution here will get deposited back on the electrode here. So this is dissolution and this is the deposition process. Okay, this is simply the two electrode setting. Now we know that EMF of cell is equals to EMF of right hand minus EMF of left hand. Now if here in this particular cell, if two electrons given by the Z and I positive because of the oxidation, these two electrons will get reduced, okay, to form the Cu here. Okay, so formation of 2+ and here because of the reduction, okay, the gain of two electrons, there is no flow of current. Means if the rate of dissolution is equals to the rate of deposition, then there will be no flow of current. So why the EMF of cell we get? Because EMF of cell is equal to right hand minus left hand. So we will get the EMF of cell. Why? Because there is always difference in the rate of dissolution and rate of deposition. And that's why the electrode potential is developed. Okay. Now, EMF of cell is equal to EMF of right hand minus EMF of left hand. Now, from these two half cells, one half cell we use or one electrode we used as a reference electrode. What is reference electrode? Whose EMF is known or arbitrarily fixed? Generally, the hydrogen electrode and canoval electrode we used as a reference electrode. Okay, isn't it? So from this right hand and left hand, one we know. When right hand and left hand attach, we will get the EMF of cell. So the EMF of other cell can be easily find out. So these are the, we can say, the various electroanalytical techniques where the two electrodes are used, and it is the conventional method. And in the UG level, we use these in the potentiometric titration. One is the reference electrode and another is our working electrode. Working electrode is also known as the indicator electrode. For example, if this is the precipitation titration under the potentiometric titration, AgNO3, KCl, we will get the AgCl and KNO3. Now you can see here, there are the two electrodes, okay? And this is simple titration. We measure the EMF of cell with the addition of the titrant from the burette. In this potentiometric titration, whatever the reaction is there, one ion must be reversible with respect to the whatever the electrode we used. Okay, so one ion must be reversible. Okay, whose concentration alters. If the concentration alters, the EMF will get changed. Okay, so if we use the AgNO3 is here, so one electrode must be Ag. Okay, so whatever the ion is there, okay, that kind of electrode we have to use. Okay, for this precipitation titration, we have to use the silver electrode. We cannot use zinc electrode or copper electrode here. So this is the two electrode system in which one is the working electrode and second one is the reference electrode. Now, this is the electrochemical cell. Now, what is electrolytic cell? Electrolytic cell is in which what happened, we supply the voltage, okay, we allow that electrolyte to decompose. Okay, so because of the electricity, there is a chemical reaction, electrolysis of that electrolyte. Simply take the NaCl, okay, take the two electrode, okay, supply the voltage. One is the positively charged electrode, another is the negatively charged electrode. Now, when the decomposition potential is reached, there is the electrolysis of the NaCl, okay, Na plus, okay, goes towards the negatively charged electrode, Cl minus negative, it goes towards the or travels towards the positively charged electrode. Okay, now Cl minus gives Cl2 plus 2 electron means oxidation. Now you can see here, this is the positively charged electrode on which oxidation occurs, but it is anode here. Okay, so anode is positive in electrolytic cell. On the other hand, cathode is negative because the reduction reaction is there in electrolytic cell. Electrolytic cell. Okay, so in electrochemical cell, you can see here anode is negative, cathode is positive. But in electrolytic cell, okay, so anode is positive and cathode is negative. Now, in electrolytical techniques, we supply the voltage in between the two electrodes and we note down the current. We measure the current. Okay, so we take an electrolyte. It is generally a electroactive material, means oxidation and reduction generally will be there. So what is the difference in between the two electrode system and three electrode system? Okay, polarography, voltammetry, amperometry, and all other techniques. These are the modern techniques in which three electrodes are used. Three electrodes are used. Now, why the polarography or voltammetry is important? Because it is applicable. Okay. Then, nature of sample, we can use any kind of nature of sample, metals, inorganic and organic material, and its loading capacity is 10 to the power minus 5 to 10 to the power minus 15. So, again, we use the aqueous solution as well as the non-aqueous solution. Now, we understand in classical techniques, we use the two electrode in which one is the reference electrode and another is the working electrode. But in modern techniques, we use the three electrode, reference electrode as it is from the classical technique, working electrode as it is from the classical technique, and another electrode is the counter electrode we used in the modern techniques. Okay, so in two electrode system, we measure the potential of the cell and current flowing through the cell. But in three electrode system, we use the potential of the working electrode, reference electrode, and the current also in between the working electrode and the counter electrode. Okay, so this is the difference. So what is polarography? Polarography is used for the qualitative as well as quantitative analysis. What kind of analyte is there and how much is both we can analyze by using this polarographic technique. Okay, Czech chemist Jaroslav Herosky won the Nobel Prize for the discovery and this invented and developed in 1922. Now, what is the basic idea in this polarography? This is a three electrode system, working electrode, counter electrode, reference electrode. So in these techniques, we apply the voltage in between the working electrode and the reference electrode. Okay, and we measure the current in between the working electrode and the counter electrode. Isn't it? So three electrodes are there, working electrode, counter electrode, reference electrode. Working electrode, counter electrode, reference electrode. We apply the voltage in between the working electrode and the reference electrode, and we measure the current in between the working electrode and the counter electrode. So this is very simple technique. What we have to understand, what is the process when the analyte is there and the three electrode system present there. What kind of analyte is there? What kind of reactions are there? How that process will be? This all we must know, and this is simply the basic idea. If we understood this, okay, analyte क्या है, उसमें क्या डालते हैं, उसके बाद में क्या होता है. Okay, so it is very simple to construct the graph. It is very simple for the qualitative interpretation and the quantitative interpretation. Okay, so we have to understand this technique. Polarography, similar like the voltammetric, all voltammetric techniques are there, chronopotentiometry, amperometry, linear wave voltammetry, and all these voltammetric techniques are used in the research field. Now, the polarography is the instrumental technique, analyze the sample qualitatively as well as quantitatively. Okay. We simply apply the voltage in between the two electrodes and we measure the current in between the other two electrodes. One is common. Working electrode is common. So the instrument is used, okay, here, apply the voltage and to note down the current. Whatever the instrument is used, that is known as the polarograph. Okay. Whatever the curve we obtain on the application of voltage, we will get the current. So current voltage curve we will get, that is known as the polarograph. And this technique is known as the polarography. So simply, we apply the voltage, but how? Time dependent. Okay, so the time dependent potential is applied to the electrochemical cell because the chemical reaction is there. Okay, and the current flowing through the cell is measured as a function of potential difference. Now, in this technique, the two electrodes, that is working electrode and counter electrode, generally three electrodes are there, working electrode, counter electrode and reference electrode, in which the reference electrode and counter electrode are non-polarizable and working electrode is polarizable. So, non-polarizable electrodes are 2 and polarizable electrode is working electrode 1. So in these polarography techniques, electrodes are there, polarizable and non-polarizable. Okay. We use the working electrode here as a drop of mercury, DMG. Okay. And negative potential is applied. Okay. So it acts as a cathode here because reduction reaction is there. Okay. Now, non-polarizable electrode, we use reference electrode as a saturated calomel electrode whose potential is depend upon the concentration of Cl- ions, and the pool of mercury, which is the non-polarizable electrode, which acts as a anode. Okay. So, what is mean by the polarization? Simply, you just consider the formation of ion, positive and negative. This is simple instrument you can see here, that is known as the polarograph, which is invented by this Herosky, and current voltage curve for the solution of 1 molar NaOH. Okay, when apply the voltage in between minus 1.9 to minus 2.0. Okay, so the negative potential is applied here. Okay, so working electrode, as already we know, it is the dropping mercury electrode. Reference electrode is the saturated calomel electrode. The counter electrode, it is also known as the auxiliary electrode. It is the pool of mercury. And we have used supporting electrolyte, and we generally use the KCl here as a supporting electrolyte. So simple experiment is there. What we have to take? We have to take the analyte. Take the analyte. Analyte is an electroactive material. It is generally the electrolyte. It is oxidized or reduced. Okay. It may be aqueous or it may be non-aqueous. Then we have to add the supporting electrolyte in it. Okay. I will explain why there is a need to add the supporting electrolyte. Then, to remove the interference of the oxygen, the nitrogen gas continuously bubbled in this solution. This is the mercury reservoir. Drop of mercury is here, which acts as a cathode. The negative potential is applied here, DC voltage. Okay, this is the pool of mercury. This is the counter electrode, non-polarizable, and this is the saturated calomel electrode. Okay, is the reference electrode. So the voltage is applied in between this working electrode or the indicator electrode and this reference electrode, and the current flowing through the cell measure in between the cathode and pool of mercury, that is DMG and pool of mercury. That is the pool of mercury is nothing but the non-polarizable electrode which acts as a anode. Okay. These are the various apparatus used for the polarography. Okay. Now, what we focused here while both electrodes, you can see here, both electrodes are mercury. This is the drop of mercury and this is the pool of mercury. This acts as a working electrode. This is the counter electrode. Now, the working electrode, the reduction reaction occurs, but on the non-polarizable electrode, the reduction reaction not occur. Both are mercury. You can see here. So we have to understand these techniques. This we are not interested in what is the diagram, what kind of the voltage is there, what kind of systematic arrangement is there. We have to understand the chemical reaction. What kind of chemical reaction, what processes are there in the polarography. So both electrode are mercury. On working electrode, the reduction reaction occurs, but pool of mercury, the reduction reaction not occurs. And this is the reason why on the pool of mercury, reduction reaction not occur. Because its surface area is very, very large. Surface area is very, very large. Okay, and that's why Okay, it formed the insoluble salt and no oxidation and no reduction occurs on the pool of mercury. Okay, now we continuously bubble the nitrogen gas to remove the oxygen because oxygen will get interfere here. We will see this later on. And in potentiometric titration, we apply the voltage up to the 3 volt. Okay. This is the dropping mercury electrode. Now, what is the difference in between the 3 electrode system and 2 electrode system? As already we know, in 2 electrode system, there is a dissolution, either a dissolution or a deposition. Means the electrode, there is a need to replace the electrode. Okay, either there is the formation of the salt or because of the dissolution or depollution, we cannot use, okay, it is not fresh every time when we perform the experiment. In dropping mercury electrode, 1 drop of mercury acts as electrode. So every time it is fresh. Dropping mercury electrode, 1 drop acts as a working electrode here, and on the single drop, the reduction reaction occurs. Every time we will get the new surface area. Okay, now why we use the mercury only? Because it displays the wide negative potential range. Okay, if we use the mercury, if itself mercury will get oxidized or reduced, we cannot use it as a electrode. But it displays a wide negative range. Okay, every time the surface area will get renewed. Okay, and it is reversible to the many kind of metal ions. And that's why we use the dropping mercury electrode. Sometimes there may be a problem, but we can reduce it. Okay, these are some disadvantages of the dropping mercury electrode. Now there are the 3 types of dropping mercury electrode we generally use: hanging mercury drop electrode, dropping mercury electrode, and static mercury drop electrode. We are interested in the what kind of reactions are in our whatever the container is there, analyte is there. Okay. So in polarography, suppose simple considered, the sample is there, drop of mercury is there, we supply the voltage, drop of mercury has the negative potential, so drop of mercury is now negative. We have take the analyte, for example, CdCl2. In this CdCl2, CD dye positive is there and Cl dye negative is there. Now CD dye positive. CdCl2 will not decompose until and unless its decomposition potential is reached. Okay, it will not attract, CD dye positive will not attract toward the negatively charged DMG until and unless its decomposition potential will reach. So we have take the analyte and we supply the voltage. We start from the negative voltage, okay, because dropping mercury electrode has a negative voltage. And in a polarography, we apply the voltage and we measure the current. Now, if CdCl2 will not get reduced, we will not get the current. You understand this? Okay, ये समझ में आ रहा है क्या आपको कि CdCl2 डिकम्पोज ही नहीं, करंट वोल्टेज पर मिलनी चाहिए. On the application of voltage, अभी करंट मिलेगा क्या? Yes. Yes or no? Yes or no?
No, current will not. It will be only.
No, current we will not get. We will not get the current. So graph पे तो कुछ भी नहीं आएगा फिर. Okay. That's why we use a supporting electrolyte. Generally, supporting electrolyte is used is a KCl. Why we use the KCl? We know in conductometry also we use the KCl to find out the cell constant. Okay, cell constant. Okay. So KCl, why we use the KCl? Because KCl will get decomposed completely. K plus ion and Cl minus ion. It is very strong electrolyte. Okay, K plus ion, Cl minus ion, its mobility is very high. Its conductivity is very high. And that's why we use the KCl as a supporting electrolyte. But in KCl, K plus and Cl minus ion, its conductivity is very high. K plus ion will not reduce on the mercury. You got it? K plus ion will not reduce. This K plus plus electron we will get K. It is not happen. It just forms an electrical double layer. When the drop of mercury comes and when it falls, okay, electrical double layer is vanished. Then again when fresh drop comes, then again the electrical double layer is formed. Okay, when drop falls, the electrical double layer is vanished. Means we will get the current when we start to apply the potential. It is because of the supporting electrolyte or the impurity present, but it is very, very less. Okay. So there are three types of current we will get in the polarography. This is the polarogram. This is the current. And this is the voltage we apply. Now, initially, from A to B, we apply the voltage, we will get the current. And this current is because of the either impurity present or because of the supporting electrolyte which we add. Now, when the decomposition potential of CdCl2 will get reached, then CD positive, okay, attract towards the or decompose and reduction reaction is there on the drop of mercury. Simple reaction is there. CD2 plus 2 electron gives CD. This is simple reduction reaction because the negative potential is applied to the mercury. A to B is the residual current because of the impurity and the supporting electrolyte. From the B, the decomposition potential is now reached. We apply the voltage, reduction reaction is there, we get the current. Again, we apply the voltage, reduction reaction is there, we will get the current. Now, the application of more and more voltage, more and more analyte will decompose on the DMG and we will get the current. Now, up to the point C, there is a linear relationship in between the current and voltage. Now, when the point C is reached, the limit is reached, when all the analyte will decompose on the drop of mercury, then further addition of the drop of mercury, the rate of travel and the rate of decomposition is same and we will get the constant current. Okay. Actually, in the current voltage curve, okay, sigmoid curve is here. We here observe only the residual current and the limiting current. And the difference between the limiting current and the residual current is the diffusion current. But in types of current, you can see the migration current also. And this migration current, we have to minimize so that we will get this sinusoidal curve. Okay, actually, this sigmoid curve is not like this. Okay, you can see here. Okay, there is a sinusoidal curve. Sinusoidal curve. Okay, why? It is because of simply the formation of drop, fall of drop, formation of drop, fall of drop, and that's why it is like this. Okay, so all these things we discuss here, how the drop of mercury comes, how the analyte reacts with the drop of mercury. Okay, once the analyte will get reduced on the drop of mercury, the amalgam is formed. And At the bottom, there is a formation of the pool of mercury, and which is the non-polarized electron. Now, that analyte, whatever the analyte is there, it will get reduced on the DMG. But how? Because of the three processes. One is the migration, second was the convection, and third one is the diffusion. Now, what is the migration? Migration is nothing but simply we know that CdCl2, CD dye positive is the positive ions, drop of mercury is the negative ions, and simply that positive ions migrate towards the negative ions. So the moment of oppositely charged ion towards the electrode, simple electrostatic attraction is there. This is the migration current. Because of the convection means because of the temperature difference, vibration is there, agitation is there, okay, temperature gradient difference is there, and that's why the convection current we get. And diffusion, in the analyte sample, okay, in the sample, the concentration of CD dye positive is very, very high. But at the drop of mercury, the concentration of analyte is very, very less. Okay, so there is a concentration difference on the surface of the DMG and in the bulk solution. The ion from bulk of solution diffused higher concentration to lower concentration. That is the movement of ion from the higher concentration to the lower concentration is nothing but the diffusion. Okay, so there are three types of process. Okay, one is the one is migration, and third one is the diffusion. Okay, residual current. We know that it is because of the supporting electrolyte or the impurity present there. The residual current is because of the formation of the condenser, that is electrical double layer, or the Faradic current. Okay, we will not go into the detail. Okay, these because of the surface tension difference and voltage difference and all. Okay, migration current, it is simply the movement of ions. Okay, and migration current, we will minimize that migration current. Migration current will minimize. How? We will see later on. Now, suppose phase of the dropping mercury electrode and will get reduced on the dropping mercury electrode. Now, there is an electrostatic attraction in between the negatively charged and the positively charged. Negatively charged electrode and positively charged. Okay, sorry, positively charged ions. Negatively charged inner cathode, that is the dropping mercury electrode, and the CD dye positive ions from the analyte. Okay, so when there is attraction in between the negative and positive, okay, the potential applied across the cathode because of these ions will get reduced and we will get the current that is simply the migration current. Now, this migration current we have to minimize. Okay, moment of ions we have to minimize because on the current voltage curve we will not have the migration current. Otherwise, we will not interpret it. There is a deviation. So, we will add the KCl, supporting electrode. What is the ratio? 100 times more than that of the analyte. So, when the negative voltage is applied, there is a migration of K plus ion and not any ions from the analyte. Okay, so the migration current, you can see here, the migration current reduced to 0 by addition of the supporting electrolyte. Okay. The second question is that why K plus ion will get not reduced? The reason is this. Okay, we apply the voltage up to the 0.5 ohm and K plus ion cannot reduce up to the 0.4 ohm. and that's why it is not reduced to the dropping mercury electron. Now, whatever the current is carried by the ions, when we apply the voltage here, you can see, okay, here, whatever the current we get, when we apply the voltage, it is because of the supporting electrolyte only. That is known as the residual current. Okay, because its concentration is very, very high. So the residual current is because of the supporting electrolyte only. Okay, and K plus ion we will not get reduced. It just form an electrical double layer when the drop of mercury is there, and we will get the current. And its concentration is 100 times more than that of the analyte. We know that the transport number, we know the transport number, okay, the fraction of current carried by the ions from the total number of ions. Okay, so fraction of current. So, as the concentration of analyte is very, very less, concentration of analyte is very, very high, the supporting electrolyte only. Okay, so its concentration is very, very high. So all the current is carried by the supporting electrolyte and not by the sample analyte because of the transport number. We know the transport number T plus, T minus and all that. KCl is the strong electrolyte. Now, what is the function of this electrolyte? Number one, it raises the conductivity of the solution. It carries the bulk of the current to prevent the migration current. Already we have discussed. It also controls the pH. Okay. And so on. Okay. So we have to use the supporting electrolyte there. Next, when the applied voltage is up to the decomposition potential or equals to the decomposition potential, then our analyte will start to decompose. There is a formation of the positive and negative ions there. And now that positive ions will decompose on the negatively charged electrode, that is the DMG. For example, CD2 plus CD dipositive gains 2 electron to form the CD, and CD reacts with mercury to form the amalgam. So the diffusion current starts. Okay, because of the diffusion, whatever the current will get, it is known as the diffusion current, and it is directly proportional to the concentration of the analyte. Okay, again, the diffusion current is directly proportional to the temperature. Increase the temperature, more and more ions from the analyte will get reduced, and we will get more and more current. Okay, then viscosity also affects the diffusion current, capillary characteristics also affects the diffusion current. Okay, then presence of suppressor. Okay, what is the role of suppressor? We will discuss it later on. The ratio of suppressor, the supporting electrolyte and analyte should be in proper proportion. The concentration of suppressor should not be more. Otherwise, viscosity is very, very high and ions will not available. We will not get the diffusion current there. Okay. Now, whatever the current we get, it is the difference in between the limiting current
Not audible, ma'am.
Hello.
Thakre madam. Thakre madam.
Sir, she
Yeah,
has
no.
left.
Madam, you are not audible, madam.
Sir, Thakre madam has left.
Why, sir?
Maybe some internet issue.
Maybe internet
Okay.
problem is there.
Maybe network issue, sir.
Okay, she might join within 10 to 5 minutes.
मुख्य जगह विपुल है यार वो तो जो के यार मैं को आपड़ था बेरियन को मतलब यार कर चुका नहीं था हड़प्पा के
Hello. Hello.
Hello, Yogita madam.
Am I audible?
Yes, madam.
Yes, ma'am.
Yes, ma'am.
yes ma'am you are clearly audible
हाँ पता नहीं आवाज को क्या हो गया
madam there is an echo problem in your side
Yes, ma'am. Now you are ready.
1 minute. कनेक्टिविटी का इशू है शायद.
Ankit sir.
स्क्रीन शेअर नाही होतंय मनीषा.
स्क्रीन शेअर नाही आतापर्यंत झालं होतं ना.
हो ते बघते ते नाही होते. Please enable access to camera.
हाँ योगिता.
Yes.
Whether screen share option is coming?
Yes. Screen share option is there, but entire screen choose what share. It is not there.
Select another option. What options you have on screen?
Chrome tab, window, entire screen. Window.
Okay.
Chrome tab.
That you can select.
मॅडम प्रेझेंटेशन ओपन करून ठेवा आणि मिनिमाइज करा त्याला. नंतर ते
सगळं झालंय ते येऊन दे राहिलं ऑप्शन.
शेअर द स्क्रीन असं काही असेल ना ते ऑप्शन.
नाही तिथे स्क्रीनच दिसत नाही, हे प्रेझेंटेशनच दिसत नाहीये.
आपलं प्रेझेंटेशन ओपन करून ठेव. मग प्रेझेंटेशन इफ इट इज नॉट ओपन.
प्रेझेंटेशन इज ओपन. Presentation is open here. It is here. is open. But in share option, it is not there on the screen. Oh,
What you will do, you leave the meeting and join again. And then you will share the screen.
त्याला वेळ लागेल भरपूर पण
ऐसे क्यों नहीं चल रहा?
Well, send then your presentation to me. I will share from my side.
रोजगार बंद हो जाएगा। मुझे देख के ऊपर आ गई, नहीं तो नीचे लेके पड़ी।
हो ना 4-5 जण थांबले।
Yogina madam, if it is not possible, share your presentation with me.
Yes, yes. Yes, I have shared. मॅडम पोहोचले तुमच्याकडे. हॅलो हॅलो कोडापे मॅडम. हॅलो
हाँ मैडम.
हाँ योगिता, just wait for 2 minutes.
होते का मी तसं पण बोलू शकते.
You will go ahead.
Okay.
I'll just share the screen. Take 2 more minutes.
Okay, okay. So the Ilkovic equation, in Ilkovic equation, the ID, that is the diffusion current, is directly proportional to the concentration. Now, from the experimental data, we will get the ID, that is the diffusion current, and once we get the value of diffusion current, the concentration of the analyte, we have the value, or we have the exact proportion. What kind of analyte is there? That concentration in terms of molarity, in terms of normality, and all that we can find out. So the diffusion current is directly proportional to the concentration. The second one, once we get the diffusion current and the half of the diffusion current means the half of the diffusion current means the only the current where the half of the analyte will get diffused, and the value where the half of the analyte will get diffused, that is known as the that potential is known as the E half. That is, E half is nothing but the potential where the diffusion current is half. That is, ID by 2 is nothing but the E half. Now we know the values of NMR and IR, and it is constant for that particular bond, particular organic material, inorganic material, whatever it is. Okay, so like that, the E half is specific. It is characteristics of that particular analyte. It may be the organic material, metal, that is inorganic material, or all kind of material. That the E half is the characteristics that what kind of material is there, and from the ID we know how much it is present. So from the ID and E half, this polarographic technique is useful for the qualitative as well as the quantitative analysis. Okay, so when we get the limiting current, it is because of the, what we can say, when all the ions from the analyte will get diffused. What we can say, the rate of travel is equal to the rate of diffusion. We get the initial current, that's the residual current. It is because of the supporting electrolyte or impurity, and the difference between these two, we get the ID. Okay, that is the diffusion current. Difference between these two, limiting current and the residual current, and the ID by 2 is nothing but the E half. ID by 2 is nothing but the E half. Now, when we construct the polarogram, in polarogram, we will get the value of ID. From the value of ID, we can calculate or we can find out the value of E half, and from the E half, we have an idea what kind of material is there, and from the ID, we know what how much quantity of that analyte is present. Okay, so I will share most of the link where you can read this material. Now, in polarography, we continuously bubble the nitrogen gas, and nitrogen gas is to remove the oxygen. Now, when the voltage is minus 0.05 volt, that if oxygen is there in the analyte, that oxygen interact or oxygen get reduced, it will interact, and there is a interact with the hydrogen present in the material, and there is a formation of the H2O2. And when the voltage is minus 0.9 volt, then that H2O2 again decomposed to form the H2O. Now, whatever the sigmoid curve we get in this particular polarography, that sigmoid curve has got two fluctuations because of the formation of the H2O2 and because of the formation of the H2O, and that's why we have to bubble continuously the nitrogen gas. That is, it is that whatever the polarography technique is there, it is free from the oxygen. Otherwise, oxygen wave will interfere the polarogram. Okay, again, one important thing that we have to not steer the solution throughout the experiment.
Whether the screen is visible, whether the PPT is visible.
Yes.
Yes.
Okay.
Yes, ma'am.
Okay. What is the
next
slide
one?
number?
Next one. Last topic up to the application. Next slide. Next. We have covered also these. Next. This also we cover. Next, residual current. Next. These are some references. Okay. Previous. Now, One important thing, we have to now steer the solution. Next, please. Next. Yes. Now this is the polarographic maxima. Whatever the sigmoid curve is there, it is not actually like this. There is a formation of the hump or this round shape. Now it is because of what? Once the drop comes, okay, the ions from the analyte, okay, try to reduce very fast, and that's why there is a hump. Okay, because the mobility of ions is very high. The rate of reduction is very high, and that's why the hump is there in the sigmoid shape curve or in our polarogram. Now, how we can reduce it? Next, please. Okay, if the viscosity of that solution will get increased, then whatever the analyte is traveled towards that drop of mercury, the rate of travel or its mobility is slow. And next, we can use these kind of material to reduce this deviation in the polarogram. Next, please. That is gelatin, dye, and surfactant. Next. These are some advantages we have already discussed. It is used for the qualitative, quantitative, single step analysis, up to the picogram and all organic samples, inorganic samples, drugs, pharmaceuticals, and so on. Next. Next. Now, how we can apply this technique for the qualitative as well as the quantitative analysis? Now, for the qualitative analysis, we know E-half is the characteristics for that particular material. Now, you can see here, suppose Cd2 plus is there, its E-half is this much. Fe2 plus is there, its E-half is this much. Now, if the value of E-half, we get the value of E-half from the polarigram, then we can find out what kind of material is there, and in this way this technique is useful for the qualitative analysis. Next. Now here this is the single sigmoid curve, this is the 2-step, and this is the 4-step. Next. And from this data of the E-half, we can have that what is present in the sample, either Cd2 plus, Pb2 plus, and so on, Cu2 plus, Zn2 plus, and so on. Okay, next. Like this, it is useful for the inorganic material. Next. Organic material, then electroactive substance. Next. We come to the next. Organic applications are there. Okay, these are the some functional group, and these are the E-half. If we get the E-half for that particular kind of reaction, then we can have an idea what kind of material is there. Next. Organic material is there. Next. Next. Now, for the quantitative analysis, that is how much that analyte is present, there are different types. From that, we can have an idea what is the quantity of that material is present. The first is calibration curve method, or direct comparison method, or standard curve method. Now, when we study the quantitative analysis, already we know what is present. How much it is present, we don't know. What is present, we know. Suppose I know that Fe2 plus is present. Then what I do, I perform a series of concentration of Fe2 plus. Okay, I will get a different type of the current. Okay, next. You can see here, suppose this is 20, 30, 40, 50, 60 milligram. We apply the voltage for these concentration and we will get the current. So we have the value of ID from this graph. In next graph, we perform the calibration curve just like our practical in UG level or PG level. So what we take? We take these concentration on the x-axis and this ID on the y-axis. And In this, we know the ID of the unknown. We don't have an idea that the concentration of that analyte. So from this calibration curve, from the ID value extrapolate, this intercept will get, we get the value of the quantity of the analyte. So this is the calibration curve method or direct comparison method or standard curve method. Next please. The next is standard addition method. Now in standard addition method, what we do, the known quantity of the standard is added in the unknown and the polarogram is taken before and after. So we know what is present. So we have the polarogram of that particular, that our analyte. Then we add the standard. The standard we add, we know its ID as well as EHA. Both we know. Again, we take the polarogram before and after the addition of the unknown and the relation is like this. Next. The relation is like this. That is the spike of sample Vs, it is a standard. Cs, it is the concentration of the original, and Vu is the unknown. And we will get this ratio. Okay. And when we simplify this ratio, this is suppose the concentration of diluted, because after addition it will get dilute. So this is the concentration from the total. What is the concentration? Just like N1V1 is equal to N2V2. This is the concentration. Next slide. And I have solved this for you. Next please, madam. I have solved this for you. How we can find out the concentration? Previous slide. So this is the last equation. If you read this Cu, next. Cu means concentration of unknown, and this is the simplification of this equation, previous slide. Okay, so you know the quantity of the analyte present. Okay, so in this way, we have an idea about the quantity of analyte present in the sample. Next. This is the second method. Next, please. Next. Previous. This is the pilot method. Now in pilot method, what we do, we know that if the relative height means the relative height of the diffusion current is same, the concentration is same. So from this idea, the relative wave height or diffusion current of two electroactive substances in the same supporting electrolyte are constant for the same concentration and are independent on the characteristics of the capillary electrode and the small temperature difference. By using this, we add the unknown. And we perform a series of known concentration values and its ID. Okay, यहाँ पे क्या करना है कि क्या है पता है, सपोज Fe है, तो Fe के डिफरेंट कॉन्सेंट्रेशन बनाने हैं, उसकी ID नोट डाउन करनी है. ओके, एंड जो अननोन सैंपल है, ओके, उसकी क्वांटिटी पता नहीं है, उसका आईडी पता है. So from the known concentration we have an ID, and when this ID is exactly matched with our sample, then that concentration is similar. So from this equation we can find out, this is the pilot method, means बहुत सारे सैंपल बनाने हैं, उसका आईडी लेना है आपको, और वो आईडी जब अपने अननोन सैंपल जो है उसके आईडी के बराबर आता है तो वो कंसंट्रेशन है अपने जो अननोन का है. सो दिस इज द ट्रायल एंड एरर बेसिस पे ये करके देखना है आपको, और व्हेन इट एग्जैक्टली मैच देन द कंसंट्रेशन ऑफ अननोन एनालाइट इज दैट. ओके, ये ख्याल में रखना है. Now, next application, there are the these are the various applications, but these few which I explain, it is from our syllabus of chemistry. That's why I explain it. Next, please. So this technique is useful for the simultaneous determination of the cation from the mixture. because ID is different, E-half is different, okay, and from that the value of ID we can calculate the concentration. This is for the quantitative analysis. Next. Determination of dissolved oxygen we can perform by using these polarographic techniques. Next. This is the equations, formation of the H2O2 and formation of the H2O. We can apply this technique for the pharmaceutical samples also. Dissolved oxygen and peroxide already we know, trace metal, then antiseptic and insecticides, vitamins, hormones, antibiotics, and so on. Next. It is having application in the pharmaceutical industry, drug analysis by the polarography. These kinds of all drugs can be analyzed by using the polarography because this is the one-step analysis method. Next. Next. In electrochemistry, polarography allows the measurement of potential. We know that the E-half, we know the E-half, and from the information, the electrode process, adsorption, desorption phenomena, we know the rate constant, and this is useful for the mechanistic study also. Next. So this is simple a polarographic technique. What we have to understand, that is this is the three electrode system. That is the working electrode, counter electrode, and the reference electrode. We apply the voltage in between the working electrode and the reference electrode, and we measure the current in between the working electrode and the counter electrode. This is simply a three electrode system reaction, techniques, in which the reaction is the reduction reaction. Whatever the analyte is there, it will get either reduced or either oxidized. Reduce means we will get, we have to apply the negative potential, it will get reduced. If we apply the positive potential, it will get oxidized. It depends on the what kind of analyte is there. Okay, so what we have to understand, we have to understand the concept of the electroanalytical techniques or any techniques. Okay, what is the principle? What should be happen? What should be happen in that beaker? Okay, analyte. What kind of reaction is there? Why we get this type of curve? Why the nature of graph is like this? Okay, why the other additives are not affect on our electrogram, or if we study the potentiometric titration, then the curve we get current voltage curve, or the potentiometric titration, the EMF volume curve, why it will not affect all kinds of the things we know when our basic concepts are clear. Basic concept is very simple. We apply the negative potential, means our electrode is negative. If positive species is there, it will attract or it will reduce on the electrode. If we apply the positive electrode, and negative species will attract toward the positive electrode. Okay, either there is a reduction reaction or either there is a oxidation reaction. Okay, in polarography there is only the reduction reaction, but amperometry is there, chronopotentiometry is there, cyclic voltammetry is there, Swift cyclic voltammetry is there, all kinds of CV curves. Okay, in amperometry, here in polarography we apply the different potential. But in amperometry, we apply the constant potential and would note down the current. Okay, so we have to understand the basic techniques behind that. All the instruments, whatever the instrument is there, either conductometry is there, either potentiometry is there, either amperometry is there, or either polarography is there, and all other. Okay, so if our concepts of the basics are very clear, okay, then we can understand all kinds of the related factors to that techniques. Okay, and it is a very perfect hand towards our experiment also. Okay, so thank you all of you. If you have a question, your questions are always welcome. Thank you.
Yes, dear participants, if anybody wants to ask, but we have, already there is 4 p.m. So I think if there are few questions, we'll take it. Couple of questions.
Yes, ma'am. I have a couple of questions. I can ask one or two.
Yes.
Thank you, ma'am. Thank you very much for the
Okay, thank you.
excellent talk on electrochemistry, particularly polarography.
Okay.
And ma'am, first question that I want to ask,
हे
why we
नको
use three?
पहिलंच लावते पहिलं ते दुसरी स्लाइड लावले मी मनीषा पहिली लाव त्याच्या आधीची हा नाव these are the three electrodes working electrode reference electrode and counter electrode.
Yes. I just want to know why we use three electrode system. What is the main purpose of three electrode system? Generally in potentiometry we use two electrode system.
two electrode system. So I have already explained कि यहाँ पे 3 इलेक्ट्रोड क्यों यूज किए. 2 इलेक्ट्रोड में क्या होता है, एक ऑक्सीडाइज होता है, एक रिड्यूस्ड होता है. जो ऑक्सीडाइज होता है वो डिसॉल्व हो जाता है, कुछ दिन के बाद वो खत्म हो जाएगा. वी कैन नॉट यूज, नया लेना पड़ेगा. ओके, सरफेस एरिया इज सेम. इफ सम इम्प्योरिटी इज देयर, वो खराब हो गया है, बहुत दिन हो गए उसको, तो वो एक्सपेरिमेंट के रीडिंग्स अच्छे से नहीं देगा. ओके, सो इट इज ऑलरेडी इन द प्रेजेंटेशन. ओके, मैंने बहुत डीपली एक्सप्लेन नहीं किया है. ओके, सो 2 इलेक्ट्रोड सिस्टम में क्या प्रॉब्लम है कि अगर कॉपर है तो बड़ा बड़ा होते जाएगा बिकॉज डिपोजिशन इज देयर. ओके, उसकी एक्टिविटी खत्म हो जाएगी कुछ दिन के बाद में. सो दैट्स व्हाई वी यूज द 3 इलेक्ट्रोड सिस्टम. इन दिस पर्टिकुलर टेक्निक the fresh surface area we will get every time. Fresh electrode we get.
Yeah, that is the advantage of the basically polarography. We get a fresh surface every time. But is there something like ohmic resistance?
Yes. So in potentiometric titration we get only the value of the EMF of cell, isn't it? And in this, when we use the 3 electrode system, we get 2 values. The EMF of cell also means what the voltage also and the current also. That is not used for the qualitative and quantitative analysis, and this is used for the qualitative and quantitative analysis. Yes.
One more question I want to ask.
Yes.
I have performed many experiments in polarography on that conventional type of polarograph, but one confusion that always remains with me there.
Yes.
Is, for example, we have a particular solution, zinc or cadmium or copper, whatever it is. For example, we take 10 power minus 3 molar zinc solution, okay, and we perform a polarograph, direct current or DPV, and we get a peak current, okay, using DME, dropping mercury electrode. And once the experiment is over, we repeat the same experiment, same solution. And after performing the experiment again, we again get the same current. For example, initially we get diffusion current, something, 10 power minus 3 micro this ampere or whatever it is. When we perform the same experiment again, we again get the same current. Although there was the concentration, because in the solution, if zinc ion is there, it is getting reduced. The concentration is reducing. And when we perform the same experiment, we get the same current. What is the cause? Because it is not the reverse. That time it is not reversible. We are not performing a cyclic voltammetric experiment. We are performing a differential pulse voltammetry or direct current voltammetry.
You are using the same analyte.
Same analyte.
Yes, we are using the same analyte. वही सैंपल आप वापस से स्टडी करेंगे तो वही करंट मिलेगा.
Yes.
No. All the zinc ion will get reduced. Okay, so we will not get. We will get the ID, but the peak height is different. ID we will get, but the peak height, the difference between the limiting, whatever the values of the residual and limiting current, it is not same. ID may be same because it may happen when
we are getting same the peak height. Whether we perform the experiment once, twice, thrice, we are getting the same peak height, means same diffusion current we are getting every time. Although zinc ions, they remain reactive with the mercury form amalgam type and all that. But that means concentration is decreasing there. The current should decrease, but why doesn't it decrease?
Yes. So what happened? ये मैंने प्रेजेंटेशन में बताया आपको कि when the rate of travel is equals to the rate of deposition, then we will get the steady current, limiting current means at the limiting value, some ion may present there. So when you perform the practical, it may happen that your total analyte will not get reduced because its concentration is very, very high. It may be less than or up to the 10 to the power minus 5. You are taking 10 to the power minus 3 means what? When the drop of mercury comes, because the reduction capacity is drop of mercury एक आएगा तो उसपे वो उतने ही आयंस रिड्यूस्ड होंगे, जितना अपना वोल्टेज आपने सप्लाई किया उतने ही होंगे, ज्यादा तो नहीं होंगे ना. आया क्या समझ में? तो
voltage is
it means
same,
that
whatever voltage we are applying. Voltage, I'm not talking about voltage, just a relation between concentration and the current.
रिलेशन वही तो मैं समझा रही हूँ कि वोल्टेज जो है आपका, तो आप जो भी आपकी रिएक्शन हुई है वहां पे, तो वो स्टेडी कब हुई? जब रेट ऑफ ट्रैवल इज इक्वल टू द रेट ऑफ डिफ्यूजन हो गया. ओके, वो क्या हो गई कि कांस्टेंट हो गई. फिर भी उसके पास में और जिंक आयंस थे, आपने वापस से वही सैंपल स्टडी किया, जो भी बचे कुचे थे वो भी वहां पे क्या हो गए? रिड्यूस्ड हो गए. अगर वो आप 2 टाइम्स नहीं, वो अगर बार बार आप रिपीट करेंगे ना, तो एक बार ऐसा आएगा कि वो आपको कर्व नहीं मिलेगी. The concentration of analyte is very, very high.
You mean there is some initially there is some saturation on the electrode because
yes, I want to say that the concentration of analyte is very, very high. You just dilute that solution 100 times and then you perform the experiment by taking 10 to the power minus 6 or 10 to the power minus 10. Then after repeating, you may not get the same. If you again get the same, you have to dilute it up to the 10 to the power minus 15 microgram. Sorry, 10 to the power minus 15 gram.
Okay, ma'am, thank you.
You have to dilute the analyte.
One more, I want to ask one more question because we are teaching at undergraduate, post-graduation level. I want to ask one question. We are using potentiometer. Is there some method for the calibration of the potentiometer? Because sometimes we are using different types of potentiometers, and maybe one potentiometer gives one reading, another gives another reading. Is there some standard method for the calibration of potentiometer?
For the calibration of potentiometer, there is actually no such method. Only we have to adjust the voltage by putting knob down 1.01 volt, 1.018 volt.
Yeah, they will do internal one, but that doesn't work always. That is why I want to
it depends on what kind of model is there. The Western bridge is there, just putting the knob down, adjust the voltage 1.018 volt. But for the pH metry, there are the method. Symmetric and asymmetric knob is there.
Meter we have, like spectrophotometer we have methods. But
pH metry we have.
Dr.
pH
Rizwan.
meter we have.
pH metry.
Dr. Yogita, I think we should stop here because
okay, thank you.
4:15.
Okay.
And
so Riyaz, then we will talk later on.
Thank you very much. Thank you for excellent.
We will share, Dr. Rizwan, we will share that. I mean, the email address and the contact number. You can just
sure, ma'am. Thank you very much. Thank you.
Yeah, whatever query you have, you can just discuss with madam later on.
Yeah, thank you very much.
So just propose vote of thanks. Whose turn it is?
Yes, already it's late. Oh, thank you.
Good afternoon, everyone. Myself, Daniel Prasad, Assistant Professor of Chemistry, PM Excellence, Government Chandra Vijay College, Indori, Madhya Pradesh. On behalf of all the participants, I would like to express my sincere thanks to our resource person, respected Dr. Yogita Thakre ma'am, for today's excellent lecture on polarography. Ma'am, your lecture was very simple, clear, and informative. We learned many new things today. Thank you for sharing your valuable knowledge and experience with us. I also thank our course coordinator, Professor Kodape ma'am, organizer UDC, MMTTC, faculty members, and all the participants for making this session successful. Once again, thank you very much, ma'am. Thank you, everyone.
Okay, thank you. Thank you. Have a nice day. Thank you.
Thank you, guys.
नहीं होते नहीं शेयर नहीं होते काय इथून आलं ते नाहीत करत अजून आलं काय झालं आपल्याला हे बघ हॅलो कुठे पीपीटी पहिले त्यांना बोलत होते ना पुढे गेलं ते सॉरी
Hello, shall I start introducing ma'am?
Hello.
Yes, ma'am. Madam, welcome, madam.
Thank you, ma'am. Thank you.
Shall I?
Yes.
Am I audible? Hello, am I audible?
Yes, ma'am. You're audible.
Yeah.
Yes,
Okay.
Deepthi, ma'am.
We can start, ma'am. Yeah. Sure.
Am I audible?
Yes, yes.
Yeah.
Good evening, everyone. I am Dr. Deepthi Verma from Department of Chemistry, University of Allahabad. It is my honor to introduce our resource person for today's lecture. We are very fortunate to have with us Dr. Anjali S. Rajput, Senior Professor in the Department of Chemistry at Dr. Babasaheb Ambedkar Matwada University. She is highly respected academician with a distinguished career in teaching and research. Dr. Anjali has over 34 years of experience in postgraduate teaching and 13 years at the undergraduate level. She has contributed 26 years in dedicated research, during which she has worked in the field of polymer chemistry, nanotechnology, and catalysis. She has published more than 102 research papers in reputed journals. As a research supervisor, she has guided more than 15 PhD scholars to completion. She has also successfully completed 5 major research projects. In recognition of her contribution, she received the Devang Mehta National Education Award for Women in Leadership in 2017. Today, she will be delivering a talk on a very important topic, development of nanotechnology, enhanced electrochemical sensing of human biomolecules, uric acid and ascorbic acid. I am sure this will be an enriching session for all of us. Let us welcome Dr. Anjali S. Rajbhoj with a round of applause. Over to you, ma'am.
Thank you, Deepti ma'am. First of all, let me thank Professor Manisha Kodape ma'am, who is the course coordinator of UGC Malviya Mission Teacher Training Center. She is from Department of Chemistry, Sant Gadge Baba Amravati University, Amravati. I would like to thank her for giving me an opportunity to deliver the lecture in this session. Thank you, ma'am. Okay. Is my screen visible? Hello?
Yes, ma'am.
Hello?
Deepti madam, you please stop your screen sharing. Deepti Verma madam, please stop your screen sharing. You have shared the screen. Deepti Verma madam. Yes. Now Anjali madam, you can share the screen.
Yes, yes.
Yes.
Can you see the screen?
No,
Hello?
No madam, it is not visible.
You have to share.
No? Okay, just a minute. Share. Okay, now can you see this
Yes, ma'am.
screen now?
Yes, it's visible.
Yeah. So today I will be delivering the lecture on development of nanotechnology, enhanced electrochemical sensing of human biomolecules related to uric acid and ascorbic acid sensing. So, this screen, just a second. I need to change it. Yes, hang it. Hello. Screen full screen. Okay. So the very can you see the slides, please? Answer.
Yes, ma'am. Yeah, we can.
Yes, ma'am.
Yeah. So let us start from the nanotechnology, the concept of it. This concept of nanotechnology was first introduced by American physicist Richard P. Feynman, called as the father of nanotechnology. He said that there is a plenty of room at the bottom. In 1959, he has said that means what he implied was that we can go to the nano level and we can work at the nano level. That this is what he meant there. Then the term nanotechnology was coined by Nori Taniguchi from Tokyo Science University in 1974. And then again, this Eric Drexler was, he popularized molecular nanotechnology. Foundation of nanotechnology. This nanotechnology is at atomic and molecular control. It is at nanometer scale, atomic size comparison. At atomic and molecular control, we are able to manipulate matter at atomic and molecular level. In nanometer scale, incredibly small scale at which nanotechnology operates, and atomic size comparison, it is relative to the size of nanometer compared to an atom. Sorry. Now, what is nanotechnology? Field of applied science whose theme is to control matter on an atomic and molecular scale. Nanometer is 1 billionth of a meter, that is 10 to the power of -9 of a meter. This is roughly 10 times the size of an individual atom. Now, how small is nanoscale? Here a comparison has been given. That is a gold atom. It is that is about 0.3 nanometer. Then glucose molecule is about 1 nanometer. Then if you compare with that of the water molecule, it is 0.1 nanometer. Hemoglobin is 5 nanometer. Then DNA, it is 10 nanometers. Virus is about 100 nanometers. Bacteria is about 1000 nanometers. Red blood cells, 10,000 nanometer. Our hair, human hair, it is about 1 lakh of a nanometer. And here an example of ant is given, that is 10 to the power 6 of a nanometer, and baseball, it is 10 to the power of 8 nanometers. Now, at the right, you can see some figures of nanomaterials which are in the range of 10 to 100 nanometers. They are liposomes, fullerenes, dendrimers, carbon nanotubes, and graphenes. So here again the comparison is given. So we'll not go into the details of this. Then I have just said that a human cross section of human hair is about 1 lakh nanometer, which is larger than nanoscale, is microscale, and smaller than it is the atomic scale. Now there are two parts of nanotechnology, that is nanoscience and nanotechnology. In nanoscience, the researchers learn about the chemical and physical properties of matter at 1 to 100 nanometer scale. 1 to 100 nanometers are called as nanostructures. They are the smallest things that can be made. Nanotechnology, in nanotechnology, the researchers develop and apply materials at this scale to develop new products or methods, that is, turning nanostructures into usable tools and applications. So here you can see some of the application, how the nanomaterials is being, we'll see this later on in detail. So nanotechnology is known to us since our ancient India. Okay, so not only in India, worldwide also, this nanotechnology is known, but at that time it was not called as nanotechnology. In European countries, you can see this cup. This is called as the Lycurgus cup. So these cups, in these cups, gold and silver nanoparticles were embedded, which cause the change in color. The red color is because of the gold nanoparticles, and the green color is because of the silver nanoparticles. And in the center, you can see the cup, which is a mix of gold and silver nanoparticles. Now you can see the Ajanta paintings also. In Ajanta paintings, the carbon nanotubes was first announced by the Japanese and was found also in the sword of Tipu Sultan. Okay, so in this, that is a sword of Tipu Sultan and Ajanta paintings, we find the carbon nanotubes. Okay, so here in the this is the picture of the ancient church in the European countries, and these colorful glasses are because of the various nanoparticles. So again, the cup is given here, which is again you can see the yellow and the red color. This is all because of the red and that is the gold and silver nanoparticles. Okay, so these nanoparticles, they show absorbance in the visible region, that is they show dark red color and also the yellow color. Now this cranberry bowl, this was also made from the made from the molten glass, and this color is because of the gold, that is gold oxide nanoparticles. So in the ancient, this cup was known, and this cup was made because of the colloidal gold. Now we also know that is the nanoparticles in our earlier century, and we consume these nanoparticles that is in the form of for increasing our we can say physics and philosophy. That is, we know that we consume gold. Here a picture is being given in which it shows that these nanoparticles are consumed by human. Because of the consumption of these nanoparticles, our physics increases, or it develops, we can say, or it becomes more robust. Not only this, our brain also becomes more, we can say, good. So here, we were consuming this gold, which is called as the portable gold, in the form of the Chyawanprash, if you remember. So in this Chyawanprash, gold and silver nanoparticles are embedded, which is good for our human health. Now, not only we were consuming silver and gold nanoparticles in Chyawanprash, we were consuming this nanoparticles also in the form of bhasmas. This bhasmas is a nanomedicine of Indian culture. So here it is an Ayurvedic herbometallic or mineral medicine used since 17th century for treating chronic diseases in very small doses. The preparation involves purification, which is also called as shodhana in Ayurveda, mixing with herbal or mineral extracts. Then the pellet is formed and incarnation to form a non-toxic ash. Incarnation means burning. So they are mentioned in ancient texts like Charak Samhita. Gold bhasma contains nanoparticles which have a size of about 56 to 57 nanometers, making it a form of natural nanomedicines. So bhasmas help in maintaining mineral balance, bone and teeth health, and body alkalinity. Loha iron bhasma is especially known for its therapeutic benefits. In the right hand side, you can see the stages of bhasma preparation. So first it is in the impure form, then it is made pure, then it is powdered, then it is triurated, means it is grinded very well, and then it is made in the form of pellets, then puta, then it is heated, and once it is heated, they are converted into the form of bhasma, and these bhasma have the size of nanoparticles. So this nano carbon tubes, which were in the ancient handicrafts and iron equipments, is shown here. So this in 1996, Robert Floyd stated that nanotechnology was used in India over 2,000 years ago in making of wood, steel, and Ajanta paintings. High resolution microscopy has revealed carbon nanotubes and cementite nanowires in wood steel. This wood steel is a unique smelting process. An addition of about 1.5 carbon produce these nanostructures, giving the steel exceptional strength and a long-lasting edge. This explains the durability of Tipu Sultan's famous sword. Nanocarbon tubes were also found in Ajanta paintings where they contributed to the material's exceptional strength and stability. Now here you can see the pictures of this that is sword by Tipu Sultan. And then you can see that at the bottom, that is the woods, that is, we can say, wood steel. Now, at extreme right, you can see nice woods. You can see the decorated piece is there. And this decorated piece is only seen in the woods, that is the wood steel. And others, it cannot be seen. And that is carved on the sword of Tipu Sultan. Now, let us come to our chemistry point of view. That when was first nanoparticles or colloidal gold nanoparticles seen? So you can say the most scientific evolution of colloidal gold did not begin until Faraday's work. Means Faraday was first to start this, and he has prepared different solutions of gold. And you can see when he prepared the different solution of golds, like from 2 to 100 nanometers, you can see there are different colors. Can you see this? The first slide, it is when he has used the nanorods, and the concentration is from 2 nanometers to about 100 nanometers. And in the next slide, you can see it is from it is also from going from the lowest concentration to the highest concentration. When it goes to the highest concentration, it becomes colorless. Okay, so this nanoparticles has the ability to show different colors at different concentration. Then Parselis' work is also known to have inspired Michael Faraday to prepare the first pure sample of colloidal gold, which he called activated gold in 1957. He used phosphorus to reduce the solution of gold chloride. Now, how strong are the nanoparticles? Okay, so nanometer sized biodegradable threads of spider silk are stronger by weight than high tensile steel. So you can imagine that this it is having just, that is, nanoparticles. If you prepare in the form of fibers, they are having very high that is strength, we can say. They are also elastic enough to stretch. Okay, so the same is the case of this nanomaterials. Now, what are the milestones of nanotechnology? Means what discoveries led to the synthesis, that is the today's developed nanotechnology? Because nanoparticles are very difficult to see by the naked eye. So we have to see it under the microscopes. So unless these microscopes were discovered, the nanotechnology could not have been discovered what it is now. So it started from 1957, where Michael Faraday synthesized colloidal golds of nanosize, which we have just seen. In 1915, Ostwald, a famous chemist, wrote a book, World of Neglected Dimensions in German, which he referred to the nanomaterials. Then in 1931, E. Rusaka and M. Knoll developed the first electron microscope. Then in 1951, E. Muller developed the field ion microscope, which enabled the imaging atoms from the tip of metallic samples. 59, Richard Feynman delivered his famous talk that there is a plenty of room at the bottom, which he pointed out to the scientists that reduced dimensionality of the materials could create fascinating materials. Then in 68, molecular beam epitaxy technique was developed. Isaki, he developed quantum size effect in semiconductors in 80. Then a quantum size effect in CDSNs, that is cadmium sulfur and cadmium selenium particles dispersed in glass, which triggered the research of nanomaterials. So in 81, scanning probe microscope was developed, that is scanning, tunneling microscope and probe. Then 85, that is fullerene was discovered, which was a groundbreaking discovery, we can say. In 89, IBM was discovered using xenon atoms. Then 1991, carbon nanotubes were discovered. And 1999, dip pen lithography, and 2000, quantum mirage using ion atoms on the copper substrate. So all these discoveries led to the present nanotechnology, which is in a very advanced form. We'll see in the later on. So these are different sizes of the nanomaterials, nanoparticles, nanorods, nanowires, nanotubes. That is single-walled and multi-walled nanotubes, nanodots, and nanoflowers. Then how to synthesize nanomaterials? There are two methods, top-down approach and bottom-up approach. In the top-down approach, we have to take a bulk material and that we have to convert into powder form. And from powder form, we have to make nanomaterials, which is shown in the left-hand side. Whereas the bottom-up approach, it is shown on the right-hand side. The molecules or atoms, they are combining, they are forming clusters, and these clusters lead to the formation of nanomaterials. Then the classification of nanomaterials, it is based on the origin, they are natural and synthetic. Based on dimension, 0 dimension, 1, 2, and 3 dimensions. Based on chemical composition, carbon-based, metals, metal oxides, organic and inorganic. Then common methods of nanomaterial characterizations, they are like, we can say, that is CLSM, AFM, SEM, TEM. These are the microscopic technique. Then the scattering techniques are DLS and SANS. And the spectroscopy are the DLS and FTIR. And the other potential techniques are XRD and DSC. So these are the synthesis methods, physical, chemical, biological, and then various other parts are given. So being a chemist, we mostly focus on sol gel, chemical vapor deposition, colloidal method, or spray pyrolysis. And also we do synthesize using biological methods, that is green synthesis using plants, using microbes also. Then what we do in our laboratory, or what I have done, how I have synthesized, or my students, they have synthesized this electrochemical reduction method. So this was this is the electrochemical reduction method. We have chosen this electrochemical reduction method because the purity of the nanomaterials which we get are highly pure in the form that is about 99.99. That is the purity of this nanomaterials which are synthesized. Okay, so you can see the experimental setup here. Here I have given here the example of nickel. We are using various other metals like copper, zinc, then various others like cobalt. Okay, so here I have the pictorial diagram. It shows the nickel plate, which is the anode. And then we are here, the cathode is the platinum plate. And then we are connecting this to a current density of 12 milliampere. And then we are dipping both these plates in a solution. This solution is TBAB and ACN, that is tetra alkyl ammonium bromide and acetonitrile and THF. So here acetonitrile and THF, this is the solvent, and TBAB, that is a tetra alkyl ammonium bromide or tetrabutyl here, we can say. In general, I am talking tetra alkyl. So that is used as a stabilizer. Okay. So then if you pass this current between these two electrodes, which are dipped in the solvent containing the stabilizer, then what happens is that the anode is a sacrificial electrode, and the ions, they start moving towards the cathode. Okay, so the here copper is shown, and in the pictorial it is showing nickel, but you can take any you can imagine anyone, because we have used many metals. So the ions from the anode, they start moving towards the cathode. As soon as they reach towards the cathode, they get neutralized, and when they get when the charge is when they get discharged. Okay, so when they get the discharge, they are trapped by the stabilizing agents, and once they are trapped by the stabilizing agent, they settle down in the form of PPT. That is the colloids you can see here. That is, they are settling down here. Can you see here? They move towards this cathode. Then they are trapped by the stabilizing agent, that is tetrabutyl ammonium bromide. And when they are trapped by this tetrabutyl ammonium bromide, they settle down in the form of colloids. Here, if you are taking a copper anode, then the dark green colored colloidal solution is being formed. The settling requires at least 2 days. Then they are washed with that is THF solution for 2 to 3 times, and they are dried in vacuum desiccators. And then once they are dried, we are using them for characterization. Now, one thing we should take very care while preparing the nanoparticles, that we should avoid the ripening of these nanoparticles. Ripening means we should not allow the nanoparticles to come close together. Once they come close together, they combine and they can form bigger particles. So this can be prevented by using of the stabilizers, and that is what we have used here, that is tetrabutyl ammonium bromide or tetraalkyl ammonium. So this is what is shown here in this, that is it is called as the Ostwald's ripening, which is prevented by the stabilizers. Now, what are the physical properties of nanomaterials? Nanomaterials, they have significantly different properties from the regular materials or regular metals, we can say, because here we are using metallic form of nanomaterials. So I'm mentioning here metals. So nanomaterials, they have less melting point, they have, or phase transition temperature is appreciably reduced, and they have less that is lattice constant also. This is because of huge fraction of surface atoms in the total amount of atoms. The mechanical property also changes. So this mechanical properties, it is about 1 to 2 orders of magnitude higher than the bulk material. This is because of the reduced defects in the nanomaterials. The optical properties of nanomaterials is also different from the bulk. The optical absorption peak of semiconductor or semiconductor materials shift to short wavelength due to increased band gap. The color of metallic nanomaterials may change with their sizes due to surface plasmon resonance. Then electrical conductivity decreases with reduced dimension due to increased surface scattering. Or sometimes electrical conductivity can also be enhanced appreciably due to better ordering of the, that is the nanostructure. Then we have the magnetic properties of nanostructured materials, which is also different from the bulk material. Ferromagnetic of bulk material disappears and transfers to super paramagnetism in the nano scale. This is because of huge surface energy. The properties of structured materials are size dependent. Properties can be tuned simply by adjusting the size, shape, or extent of agglomeration. Now we are coming to our that is topic, that is nanotechnology in electrochemical sensing. So when we are using these nanomaterials, they have increased surface area. So when they have increased surface area, there enhances sensitivity due to more interaction sized. Enhanced electron transfer, this is because of it improves the selectivity for specific biomolecules. Then there is more functionalization because it increases the selectivity for specific biomolecules. Then metal nanoparticles offers conductivity and catalytic property. Carbon materials, that is nanomaterials, provide high surface area and electrical conductivity. And metal oxide enhances electrochemical properties of sensors. So here we are using the last one here, that is the metal oxides or metal nanoparticle, the third one, the fourth one, sorry. Here, okay, because we are using the metal nanomaterials. Now, this synthesis, so we are going to synthesize two nanomaterials, monometallic nanomaterials. First is the zinc, and the second one is the copper. So here the synthesis of zinc nanomaterials is shown. This you can see here, the electrochemical method is used, which I've just explained to you. That is this figure. Yeah, the same method is used for synthesis of this. That is the zinc nanomaterials. So here the zinc metal, it is sacrificial anode. Platinum as usual is the cathode. Then we are using capping agent and solvent, and then continuous stirring, constant current was applied, that is 12 milliamperes, and settling for 2 to 3 hours, and then washing with THF and drying in the vacuum desiccator. And then we are using also for this calcination. So this is what is shown, what are the reactions that are happening at anode and cathode. So which I have already explained to you. So we'll not go into the, now let us come to the characterization point. So this is we have taken the ultraviolet that is absorption, that optical absorption spectroscopy, that is UV visible spectroscopy we have taken. So here you can see here this zinc oxide nanomaterials, they show the absorption in the UV range, that is about 277.6. And you can see here we are using here water. We are using water acetonitrile. Then we are using here two surfactants, that is you can see here tetrabutyl ammonium bromide, TBAB, and here we are also using TPAB, so tetrapropyl ammonium bromide. So these are the four different types of that is zinc oxide nanomaterials we are showing here, and the absorbance is slightly different. So next is the FTIR spectrum, a spectrum of that is zinc oxide nanomaterials. So here we can see the first sample is before calcination, and the next below it is after calcination. So you can see before calcination, we can see the OH stretching free peaks for both A and B. Then we can see the CN linkages, which is at 1031 to 1087 per centimeter, and sharp peaks are that is around 410 to 541. Okay, that is in the range of 400 to 500. So this is this range, that is 400 to 500. This indicates that our zinc oxide nanomaterials are being formed. Okay, so after calcination, what happens is that the OH peaks, because we are calcinating it around 500 degrees Celsius. So the OH peak, it disappears. Okay, and there are no peaks shown, that is green colored. Can you see after calcination, there are no peaks which are seen because we are heating after that is 500 degrees Celsius. And also the peaks of CH stretching or conjugated CN linkages or NH, they also disappear. And the peaks we are getting, they are of only that is zinc nanomaterials. Okay. Next, this is the XRD pattern of zinc oxide nanomaterials at different reaction parameters. So here we can see the peaks at 31. The peaks which you are getting, they are 100, 002, 101, 102, 110, and 103, 112. These are shown in the XRD spectrum, your pattern you can see here, and they are also matched with the JCPDS file. Okay, so here we can say that the shape of these nanomaterials are hexagonal wood side face. Okay, so we'll rest will not, and the size we can say here, it is given at extreme bottom, that is it is 46.5, this 37.3, 50.8, 53.9, and 31.5 nanometers. These are the size of the nanomaterials. Now this same analysis we have done, and in this same analysis we can see here that spherical and rod shaped zinc oxide nanomaterials, they can be seen. This is the change in the shape, this is because of different reaction parameters. Then the EDS analysis is seen here. Here the percentage of atoms is given. So the oxygen percentage is about 82.07 and zinc it is about 17.93, and if you add this it becomes 100. So from this spectrum we can say that no other impurity is being present in this our synthesized zinc oxide nanomaterials. Then this is HR10 and a said pattern is also given here. So if from this TEM, that is transmission electron microscopy, we can calculate the size, which is about 50 nanometers. That is the here, and mean size is also given here. That is about 34.44 nanometers. And here, whatever the peaks we see in the XRD data, the same peaks can be seen in the said pattern. You can see here the extreme right at below. The said pattern shows different peaks. The same peaks can be seen in the XRD pattern also. And this we can say that we are getting so clear peaks. It means our synthesized nanoparticles of zinc, they are crystalline in nature. Here also the same is shown here. This synthesized zinc oxide nanoparticles, they are polycrystalline in nature. So the said pattern and XRD, they are in good agreement. Then these are the effect of reaction parameters on size and shape of zinc oxide nanoparticles. If we have seen different reaction parameters, that is the effect of current, effect of solvent, effect of capping agent, and effect of calcination time. So here it is being observed that if you increase the current, you will find that the size, average size of nanoparticles, it decreases. Here capping agents also, if the length of the capping agent is more, you will find that the size of the nanomaterials is decreased. So, and also the effect is that we have different shapes of nanomaterials, that is flower shape, they are there, then sheets are there, spherical are there, then rods are also can be seen, and flowers can be seen. Now this is again the same synthesis of copper nanomaterials, the same method we have used here, so I'll not repeat here. So this is the visual inspection of this copper oxide nanomaterials which are formed, which are dark green in color. And here this shows a UV visible spectrum at, that is 642 nanometers and 639 nanometers. So this is that is the absorption spectrum of copper oxide nanoparticles. Then FTIR analysis is being done of calcined and non-calcined copper oxide nanoparticles. So whatever the hydroxide peaks can be seen in non-calcined spectrum, they disappear after calcination because we are heating the copper oxide nanomaterials for more than 500 degrees Celsius. Okay, so all these peaks disappear. So only the peaks corresponding to, you can see here in this case, this or this, it is at 713 and 758 per centimeter. They correspond to copper oxygen stretching, which are attributed to the formation of copper oxide nanoparticles. Then this is the XRD pattern, and we can see here, these are the different peaks which are shown here. Okay, so here average size is calculated here, which is about 0.4. And then you can see here that is here also average, average size of copper oxide nanoparticles at different current densities being shown here. This is calculated from XRD data. Then the same analysis is shown here. So you can see here the copper oxide nanomaterials, they are flower-like shapes, some are spherical and all. You can see this same images were carried out at current density of 8 milliamperes and 12 milliamperes. Now this EDS spectrum is also shown here. We get a percentage of only copper and oxygen, which is about 100%, which indicates that copper oxide nanoparticles which are formed, they are highly pure in nature. No other traces of any element can be seen here, which indicates a highly purity of our nanomaterials. Then the transmission electron microscopy, it is shown here. So here we can see this size, it depicts the size that in TEM, and here also the said pattern is being shown, which matches with the XRD data, and the mean size, it is about 14 to 15 nanometers. Now then we have synthesized the bimetallic nanomaterials. Why bimetallic? Because they have high surface to volume ratio to enhance optical properties, dielectric properties, sensing properties, to enhance mechanical strength and develop new smart materials for use in modern and future technologies. So here you can see here we are using the same electrochemical reduction method for synthesizing the bimetallic, that is copper, zinc nanoparticles. The same we can say experimental is used, but here what we are keeping is that two anodes are used here, that is copper and zinc, and in between these two anodes we are keeping the cathode, that is the platinum, and in this way we can synthesize the bimetallic nanoparticles. So in the UV visible spectroscopy, you can see the two peaks are there. One is at 367 and 360. So here, and one is in the UV region. So the 360 and 367, these peaks corresponds to copper, whereas in the UV region they are corresponding to the zinc, which slightly can be seen on the extreme left hand side of the graph of UV visible spectroscopy. Then there is a visible spectra of copper. You can see these bottles are shown here. The material is settling down. This settled material is of copper zinc nanomaterials. And here this is the sample bottles which are shown. In the FTIR spectrum of copper zinc bimetallic nanoparticles, you can see the peaks. And we are only interested here is in the peaks which are at 400 to 800 per centimeter, which corresponds to metal oxygen stretching bands and which indicate that copper and zinc bimetallic nanomaterials have been formed. Then the X-ray diffraction analysis, here you can see the peaks. That is both peaks of copper as well as zinc are shown here in this X-ray diffraction spectrum, and which that is the copper oxide nanoparticles. They are monoclinic in nature, whereas the zinc oxide they are having hexagonal phase. Okay, so these two are confirmed, that is the formation of copper oxide and zinc oxide bimetallic nanoparticles were identified and matched with JCPDS, that is 36-1451 database, which confirms that the bimetallic nanoparticles of copper and zinc oxide are being formed. Now, estimation of crystalline size. The crystalline size of our bimetallic nanoparticles were like 34, from like 25 to about 35 nanometers, which can be shown here. Then in the scanning electron microscopy, so you can see that they are spherical in shape. The EDS indicate that the formation of pure copper zinc bimetallic nanoparticles was successfully prepared. Then EDS, it shows that no impurity is being present. We are getting peaks only of copper, zinc, oxygen, and the total is like 100. So we are getting 100% pure nanoparticles of copper and zinc oxide. Then this is the TEM and SED analysis. So particle size is about 20 to 50 nanometers. Then we are that this SED patterns, the concentric rings of this SED pattern, that shows that our nanoparticles are crystalline in nature and their size is in agreement with the XRD analysis. Then we have done the sensing study, and this sensing study we have done on electrochemical measurements. That is here. This we are using. We are studying on this electrochemical measurement, the cyclic voltammetry, differential pulse voltammetry, and square wave voltammetry measurements. This were performed on Medtrome Autolab PG Stat. One compartment, it is a one compartment. You can see it here that this is the box, and here there is the electrodes are there, and these three electrodes consist of saturated silver chloride, which is called as the reference electrode, then platinum electrode, and modified platinum glassy carbon electrode as an working electrode. So electrochemical here, sorry for this, it is not dopamine. This is, we are doing the studies on uric acid and ascorbic acid. So measurements were carried out in 0.1 molar PBS at room temperature. The CV, DPV, and SWV were done at various concentration in the range of 50 micromolar to 350 micromolars at 100 to 100 millivolts per second. This is the scanning range we are taking here, and the electrochemical behavior of bare glassy carbon electrode and metal oxygen nanoparticle glassy carbon electrode were studied by cyclic voltammetry in 0.1 millimolar ferricyanide solution at a scan rate of 100 millivolts per second. Okay, so here this is the instrument we are using here. Now, first we have to fabricate the electrodes. So how to, that is the glassy carbon electrode or glassy carbon or platinum electrode has to be modified. And what modification means, we have to coat our nanoparticles on that electrode. So here it is given how we coat this. First we are taking zinc oxide nanoparticles, then we will be taking copper oxide nanoparticles, and then we will be taking the bimetallic copper and zinc oxide nanoparticles, and we will be coating on the electrode. So how the coating is done, that is 2 milligram of zinc oxide nanoparticles were added to 2 milliliters of 0.1% Nafion alcoholic solution, and then it is sonicated for about 20 minutes with an ultrasonicator to get a stable and homogeneous suspension. To prepare glassy carbon or platinum modified electrode with zinc oxide nanoparticles, an alcoholic solution of 0.1% Nafion dispersion on zinc oxide nanoparticles was prepared, and suspension was cast on the surface of the pre-treated platinum, gold, or glassy carbon electrode. The solvent was allowed to evaporate at room temperature for 2 to 3 hours, which resulted in immobilized zinc oxide nanoparticles on the electrode surface. And then the electrode was washed several times with double distilled water and kept in air. Uniform black film was found to be formed on the platinum or glassy carbon surface, and this was used for our further studies, that is the sensing studies. Now, first thing we are using is the ascorbic acid. Now, why we have used ascorbic acid? Ascorbic acid, another name is vitamin C, which is an essential nutrient of your body, which cannot be produced in our body. It drives critical functions like synthesizing collagen for skin, joint health, neutralizing cellular damage as an antioxidant, boosting immune function, and significantly enhancing the absorption of iron from the plant-based food. It is also known, I will not go into the details of this ascorbic acid, but we'll just say that here that it is used for tissue growth repair, then it is used for cell protection, iron absorption, immune support, and preventing scurvy. The detail is given here, which you can see later on in the PPT, which I'll be sharing. Okay. So here, you find here that ascorbic acid is a very vital, we can say, nutrient for our human health, and therefore we have used this ascorbic acid for sensing purposes. So here you can see the first curve which is shown is the electrochemical, that is the cyclic voltammetric curve, which is shown. First curve is of bare, that is platinum electrode, and which is shown with the blue line, and the red one is the modified platinum electrode. And then we are in the next, you can see that this is at different scan rates. You can see we are varying the scan rate like 25, 50, 75, 100, and 125. And then we can we are taking the and plotting it here in both these two graphs which are shown at the bottom. So it was seen that the electroactive surface of modified that is here, it was found to be 2.05 times higher than that of the bare electrode. The platinum electrode show reversible behavior in non-reversible redox conditions. This here we are using, first of all, we are using here the copper one, and here we are using copper platinum electrode. Okay. So next, these curves which are shown here. They are ascorbic acid molecules that have been adsorbed on the surface of copper platinum electrode with the help of water. The ascorbic acid molecule is hydrolyzed and oxidized to dehydroascorbic acid. This oxidation of ascorbic acid can liberate electrons that are associated with detectable current. Lack of reduction of Lack of reduction peak. We cannot see the reduction peak here on the that is spectrum, or we can say curve. So there is we can say that there is no reduction of the ascorbic acid. Okay, so both these curves which I am showing here, we can see only the oxidation curves. We cannot see the reduction curves here. So that means the ascorbic acid can only be oxidized, and if you want to reduce it, cannot be reduced. So there is only one peak that is seen here. So this here, the result for modified copper platinum electrode required a significant peak at ascorbic acid absorption of 0.5 volts. Now these are the anodic peaks current found linear to the ascorbic acid concentration. So the correlation coefficient for linear regression equation, this is equal to 0.032x minus 12.3 was observed to be 0.1933. Okay, and sensitivity was found to be 9.33 milliamperes. So these are the different curves. That is a linear regression. This is the curves for difference pulse voltammetry, and here also we detect the sensitivity. It is about 1.65 microampere. Then this curve is for square wave voltammetry. All these curves show us the sensitivity. So here the sensitivity, it is found to be 2.89 microamperes of ascorbic acid. Then we have also studied for that is the zinc oxide, that is zinc nanomaterials. And here also you will see that we can say the correlation coefficient of it was equal to 0.981. So only the oxidation curves are seen here for using the zinc oxide nanomaterials. No reduction peak can be seen here. And this is the graph which is of differential pulse voltammetry and the square wave voltammetry. The down one is of square wave voltammetry, and in which both these graphs show us the sensitivity. In differential pulse voltammetry, the sensitivity was found to be 0.552 microamperes, and in square wave voltammetry, it was found to be 0.85 microamperes. So here, both show that the sensitivity was found to be good, and if the sensitivity is good, we can detect a very trace amount of ascorbic acid found. Here, this is what is the comparative study of various electrodes. Then the uric acid for, we have also used the uric acid for human health. Now, why we have chosen the uric acid for uric acid is that as a primary end product of purine metabolism in human body, 2,6,8-thiohydroxypurine, often termed as uric acid, is present in different biological fluids such as urine, saliva, blood serum, and plasma. Important biomolecule uric acid is released from the human body and is frequently seen in blood, serum, urine, or biological fluid. Uric acid can crystallize as urate when it is present in excess in body fluids, which can result in gout or kidney stones. If it is present in excess, the human body can have kidney stones or gout. What are the benefits of uric acid? It is antioxidant protection. Uric acid accounts for overall half of the body's total oxidant capacity, defending nerve tissues from free radical damage. Neurological support, balanced levels are also associated with reduced risk of certain neurodegenerative conditions. If high uric acid is present, it is called as hyperuricemia, which results, as we have discussed, in gout, which is excess uric acid crystallizes in joints, triggering pain and swelling. And then kidney stones are formed in the kidney. So this study is also done using copper nanoparticles. So here we can see in this first, we can see both the oxidation as well as the reduction peaks are seen. And then we have taken the slope of this and we have plotted it here, and the scan rate is measured and square root of scan rate is taken. So this is what is shown in this cathodic and anodic peaks, which are shown in the first graph. They are linearly proportional to the potential sweep rate, demonstrating the uric acid concentration is surface control in this range of potential scan rate. Then here we can see the peaks which are shown, that is the oxidation as in reduction peaks. This is of first is the bare platinum electrode, and the second one is red one is red one is bare platinum electrode, and blue one is the modified platinum electrode. And here these are the different this is at the different scan rate, and then we have plotted it here. Okay, so here linear plot is done of the peak current versus concentration of uric acid. So here this peak of cyclic voltammetry, you can see first it increases. It increases with increasing in the uric acid concentration. The results reveal that the peak currents of potential around 0.53 volt steadily raised with increasing uric acid concentration. The calibration plot for peak current versus uric acid concentration was produced using a linear regression equation with R square is equal to 0.948 and R square is equal to 0.991, and the sensitivity was found to be 0.828 microamperes and 0.606 microamperes. So this is the electrochemical uric acid determination of zinc platinum electrode. So at different concentrations. Then this is for the differential pulse voltammetry, that's electrochemical detection of uric acid by differential pulse voltammetry, and then below we are detecting by square wave voltammetry. So both we are getting a very good sensitivity. In the differential pulse voltammetry, we are getting a sensitivity of 0.1086 and 5 microamperes. So we are detecting it by, now here we are taking, you can see here we are taking bimetallic copper zinc nanoparticles. Both copper and zinc, they are used here, and we are studying the uric acid here, and we are getting both the oxidation as well as the reduction peaks in the cyclic voltammetry. And here that is sensitivity, it was found to be 0.38 microamperes and 0.31 microamperes. Okay, so in the here, then this is by differential pulse voltammetry, the sensitivity is 0.54. Then this is the comparative studies of copper, zinc, and copper zinc bimetallic nanomaterials, and here the sensitivity data is given here. So you can see here we are getting very the sensitivity which we are getting here, it is in point. Okay, so this means our that is the sensing study, it is it is applicable if traces amounts of that is ascorbic acids and uric acid is being present. So this was all regarding our that is the research work. Is there any questions regarding anybody asking about this research work?
Yes, ma'am. I have a question.
Yeah, you can ask. This is all regarding my research work, and you can ask me after completion. But I just wanted to hear whether you are attending my lecture or not, because I have simply been talking and there is no response from your side. So that is why you can ask me after this my session is over. Now let us see the applications which are found in the nature. Okay, so this is the nature, how it works at nano level. Okay, so if you see here, here an example is given of a lotus leaf. Okay, so we all know that a lotus leaf, it is having a very high hydrophobicity. That is, we say it is having a super hydrophobicity, that is repellence for water. Now this repellence, because if water falls on this leaf, you'll find that it does not stick to the leaf, it rolls off the leaf. Okay, so this if you see the leaf under the microscope, you'll find that there are some bumps on the leaf of the lotus, and this this phenomena, this bumps which are there on this lotus leaf, they are nanometer size. This helps the leaf that the water can go or fall off without spreading on the total leaf. So water drop falls on from the leaf and it collects the dirt along with it, because lotus is generally found in the we can say lakes and where there is a lot of dirt. Okay, so there is self-cleaning properties there of this leaf of the lotus, and this self-cleaning properties was used by our human being to make water repellent clothes. Can you see here on the extreme right, there is a water repellent clothes. This is how we are making the water repellent clothes. Now we are also making nanomedicine. Earlier, a lot of medicine used to be given, a lot of pills were given. Now, since nanomedicine is being available, the number of pills has been decreased. So why? Because nanomedicine is being used to cure diseases, repair damaged tissues such as bone, muscles, and nerves. Then nanomedicine, it ranges from medical applications, then nanoelectronic, nanobiosensors, and even possible future applications of nanotechnology. So this nanomedicine, it is not only used for diagnosis, it is also used for therapeutic uses also. So diagnosis means for imaging, that is the MRI scan or whatever you do it, no. So at that point also we are making use of nanotechnology, and also therapeutic means delivery of medicine to the exact location, killing microorganisms and cancer cell and cell repair. So here the nanomedicine is being used. Now here you can see it is also used, that is silver nanomaterials. They are also used for healing burns. So a new skin gel made of silver nanomaterials may help improve the treatment of burns. So mostly the gels or the antiseptics which are used for treating of the burns, they are silver based, and they contain sulfadiazine and sulfur nitrate. So these are germ fighters and they save lives and speed healings. So gel, it is composed of that is 1 by 50,000 the width of human hair, that appears more effective than traditional gels. So this is what is being used here. Not only this we can see here in this figure, the nanoparticles are nowadays used for cancer treatment. Tumors have large interstitial spaces in them, and in this large interstitial spaces of tumor, the nanoparticle can penetrate into it and they can destroy this tumor. So here an example, can you see this? The white white are nothing but the nanoparticles. They are mostly of gold. You can see here yellowish nanorods, nanocages, and clusters are shown here. And this red is the tumor which is shown here. So nanoparticles can penetrate into this tumor and they can destroy or kill the tumor from inside, and also they can kill from the outside. Okay, so this is what is shown here. So nowadays, these nanoparticles are used for cancer treatment. Then now nanoparticles are also used in cosmetics also. That is micelles. We all know that micelles, they are amphiphilic in nature. They are having, that is hydrophilic as well as lipophilic part in them, and therefore they are called as micelles. And they are here, they are very small. That is, this is the example of micelle which is given here. So these micelles, because they are having hydrophilic as well as a lipophilic part in them, they can be used as cleansers in our washing. They are used to remove dirt particles. They are also used in makeup and removing excess sebum from the skin by enveloping the lipophilic material. And then they are used for the cleansing purposes also. So this is a diagram you can see here. That is the micelle, and this is the this is the cross section of the micelle. Then this is the buckyball, which is called as the fullerene, that is C60. That is it is also called as Buckminster fullerene. It is a iconic nanomaterials. It is about 1 nanometer in diameter, and it resembles a soccer ball. So this that is fullerene. It is used in cosmetic industry as a black coloring pigment, and it is characterized by particularly high color density, intensity, and depth. It is found in mascaras, eyeshadows, eyebrow pencils, and since the beginning of European Union cosmetics legislation and is used worldwide today. It's applied in highly pure form and is subject of strict quality requirements. This is, you can see the fullerene molecule here. Then it is also used in agriculture. So controlled release of nanofertilizers for productivity, nano-based target delivery approach for gene transfer that crop development, nanofertilized devices providing smart monitoring and nanomaterial plant stress tolerance and stimulant parameter. Then there are the nanopesticides I've just mentioned. So these increased efficacy, durability, and reduction in amount of active ingredients easily penetrate into the that is plant, and they can destroy the insecticides, which materials are used in bulletproof vests, body armor. So here, this why it is being used, that is we are using here tungsten and not carbon. Tungsten nanoparticles. We are using tungsten nanoparticles because they are super strong materials. They are 5 times stronger than steel and at least twice as strong as any impact resistant material currently in use as protective gear. So it is very much strong. It has withstood of dropping 4 diesel. Okay, it is you just understand this, that they are very strong material, that is tungsten nanomaterials. So they are made, they are applications of this tungsten nanomaterials are, they are ballistic protection, personal body armor, bulletproof vests, vehicle armor, shields, helmets, and protective enclosures. So here you can see the pictorial diagram of this. That is not only the bulletproof jackets, they are light, because if it is heavy, then it would be very much it would not, or they are made light, but they are bulletproof. Okay, so here no bullet or sharp nail can enter this bulletproof jacket. See, this is the mechanism of this. When the picture which is at the top, it is a normal material, glass material. It breaks when the bullet strikes it. But if the material which is shown at the bottom, you can see it is like 2 to 3. You can say slide given of this material here. So you can see this material is made of polycarbonate. It is also made of tungstate. 3-4 layers of this tungstate are given. So in the second picture, if you see the bullet strikes this material, so it does not penetrate into that material. Now this material, this is another material which is used here, which is made up of polycarbonate. You can also use the tungsten nano materials also. So this is a mechanism of nanomaterial used in bulletproof vests or jackets. Now this silver nanoparticles are nowadays, they are used in ink, board, chip circuits. The circuits are not only available in our television nowadays, but they are used in our laptops, they are used in our computers, they are used in our mobiles also. So the researchers, they have used silver nanoparticles, ink as bonding agents. Since the metal is easy to come by and process, only requires a very small amount, keeping the cost low. So bonded circuit chips called surface mounting devices and electrical components that are placed directly on the top of printed boards. They are used in complex electrical system, such as I have told you, computers, mobile phones, to enable specific functions and deliver high performance. Then this is our last slide. You can see here, I'm using this, I'm showing you here the feather of peacock. You can see that we can see many color of this peacock feathers. Now, as the angle changes, you will see that the color of that feather, that is changes of the peacock feather, it changes. So this is because it contains internal structure. It contains tiny barbules, and that produces the hue. Hue means change in color. This coloration is because of internal reflections, repeated structure of parvules similar to the way of lattice arrangement of the crystal. So this is also one form of the nanotechnology. This feather, it contains natural nanotechnology that has potential to transform optics when this remarkable approach is adopted for use of human technology. Many a times we are using this technology for preparing different colors also. Okay. So thank you. And now I'm most welcome for your questions.
Yes, good evening, madam.
Good evening. Yeah.
Yes, madam. Thank you, madam, for your wonderful lecture about the topic nano materials in electrochemical sensing.
Yeah. I
have a minor doubt. What are the capping agents you have used, madam, for the synthesis of these nanoparticles?
I have used tetra alkyl ammonium bromides. In alkyl, I have used like tetra butyl ammonium bromide, tetra propyl ammonium bromide, tetra pentyl. Anything I can use here, up till octyl.
Madam, but in all the characterization techniques, they have not shown, madam, anything, and you are telling that the nanoparticles are very pure.
They are.
Why these capping agents are not shown in analysis, madam? In the
no, we can no. Actually, this is because we have calcined that material. Can you see this graph? Yeah. These just a minute. I'll show you.
Well, actually, that
can you see this graph? Just a minute. Can you see this graph? IR graph.
Yes, madam. Yes, madam.
This is non-calcined copper oxide nanomaterials, and this is the calcined. So if you see the non-calcined copper materials, you will see there are many peaks. This OH peaks, which is shown at like 3000. This I'm showing with the round. This hydroxy peak is because of the capping agent, which attracts the water molecules, and it is shown here. But when we calcine or when we heat about 500 degrees Celsius, this peak disappears. You can see in the IR, there is no peak observed here. Okay.
Yes, madam, but actually the capping agents are added to stabilize the nanoparticles, isn't it, madam?
Yes.
But after calcination, how they are stabilized, madam?
They are stable because we have there is no trace of water molecule, no, so they cannot agglomerate.
Okay, madam. Okay. Thank you very much, madam.
Yeah.
Hello.
Any other question?
Yes, ma'am. Two basic questions.
Can you talk about the ISIL's nanoparticles?
Sorry.
Penetration of cosmetic ingredients.
So I cannot hear you. You are not audible.
Madam, I am audible. Hello.
Yeah, you are audible. Yes.
Madam, what do you do to prevent that?
Sorry.
That वो जो नेफ्यून मेम्ब्रेन फट जाता है उसको रोकने के लिए क्या करते हैं आप?
No, I can't hear you. Sorry, I can't hear. You are not audible. Your voice is not clear.
Hello. मैडम आ रही है आवाज?
हाँ, ठीक है.
मैडम जो नेफ्यून मेम्ब्रेन ड्राइंग के दौरान जब वो फट जाता है उसको रोकने के लिए क्या करता है? उसमें जो क्रैक्स डेवलप होते हैं उससे रोकने के लिए क्या करता है?
क्या हो जाता है?
नेफ्यून में क्रैक डेवलप हो जाते हैं ना ड्राइंग के दौरान जब ड्राइंग कर रहे होते हैं.
Can you see here? We are using only 0.1% of Nefian solution. Can you see this slide?
Yeah.
0.1% is used here. So did you use the Nefian solution? The concentration was higher.
Yeah, we tried, but
no, the concentration should be less. And it is. Can you see here? We are using only 2 ml of 0.1% Nefian alcoholic solution.
Okay.
And then it is sonicated to about 20 minutes, and then we are putting it on the electrodes.
Okay.
So concentration, you look for the concentration.
Okay. And one more question: how do you maintain that constant current density? Because anode is sacrificial anode.
No, current is constant. We are putting it to a source where it gives you a constant current. Here it is not shown in the picture, but actually we are using a power supply which provides current in milliamperes, and that source is giving you constant current.
क्योंकि एनोड तो ग्रेजुअली खत्म हो रहा है, सेग्रीगेशन हो रहा है,
तो फिर दूसरा ले लेना वो खत्म होने के बाद।
तो मतलब सरफेस एरिया कम होता जा रहा है, तो फिर करंट भी साथ साथ कम होता जा रहा है।
नहीं, करंट बिल्कुल नहीं कम होगा, हम करंट सप्लाई कांस्टेंट रखना है।
मैडम फिर करंट का वो था ना, एरिया करंट डेंसिटी थी, तो डेंसिटी जब एरिया कम होता जा रहा है, करंट डेंसिटी कैसे कांस्टेंट मेंटेन?
नहीं कांस्टेंट, दैट इज नो रिलेशन बिटवीन द एरिया कम हो जा रहा है और करंट डेंसिटी। करंट डेंसिटी उतना ही रहेगा, वो एनोड जो है वो कम होते जाएगा और अल्टीमेटली इट विल आफ्टर 2 to 3 years इसका साइज कम हो जाएगा, और जैसे उसका साइज बहुत ज्यादा कम होता है हम रिप्लेस करते हैं उसको दूसरे।
Okay.
The point here is you should keep the current constant current. The current should not be variable. We have a lot of instruments where they give you a constant current.
Okay, thank you.
Yeah.
Is I audible, ma'am?
Yeah, now you're audible.
As you say that the micellar nanoparticle nuts. So the micelle enhances the penetration of cosmetic ingredients into the skin.
Yes.
So how do we as a researchers ensure that the beneficial molecules are delivered effectively while whatever the that means while preventing the unwanted penetration of potentially hazardous substances?
So now whatever I've shown you this slide, this is a general slide. Okay, so in which the micelles, they are used in cleansers. अपने जो face wash होते हैं, body wash होते हैं ना, उस cleansers में या toners में use किए जाते हैं micelles. So they